Hello, Karine.
No sound. Okay. Can you hear me?
Yes, bonjour,
Hello.
Karine.
Hello, how are you, Dalian?
Nice, thank you. So good to
Good.
see you here with
Yes,
us.
yes, we're
We are
good.
we
Yeah.
are just waiting
Okay,
to start in the platform, but
good.
we will start soon, okay?
Yeah. Do you want me to try just the sharing, just to test?
Yes,
Okay, I
let's
can
go.
try to test just to be sure it's working well.
Okay.
Okay, so
Uh,
yeah, can you see?
Not, but let me check.
Not yet.
I guess I have to stop my sharing here. One
Ah, okay.
second.
We can't see it from here, Karine.
Ah, you can see. Okay, perfect.
Yes, is it okay?
Yes, and Dalian, you can see it.
Let me see why I can't see. Yes, I can see now. I'm
Okay,
sorry.
perfect. Okay. So I
Great.
can stop. Okay, perfect. So it's working. So I will share as soon as it's okay.
Okay. And now I am sharing my screen.
Yeah, we can see it. Yeah,
Great.
okay.
Adma, please let us know when we can start, okay?
If you want, you can start. Is everything okay with the platform?
Okay. So it's 9:03 a.m. and we are going to start. So good morning, everyone. It's a great pleasure to welcome you all to the second scientific meeting of Immunometabolism. We are absolutely delighted to have such a distinguished group of research and students join us today. This event holds particular significance as it is a core activity realized under the umbrella of our ongoing international collaborative projects, notably the important work founded through the CAPES-COFECUB and CNPq international programs. We are proud to host this meeting as a demonstration of the synergy and progress achieved through this essential partnership. Before we begin our scientific program, please allow me to provide a quick guidance regarding participation. We encourage everyone to actively engage with our speakers. To ensure a smooth flow of presentation, we kindly ask that all questions be submitted through the chat. All submitted questions will be compiled and addressed by the speakers immediately following the conclusion of their presentation. Thank you for your cooperation. We wish you an insightful and productive meeting. Let us begin this section. I will start by sharing some of the joint study we have undertaken over the past 10 years. This collaboration is based on our common goal to deepen our knowledge in the fascinating field of immunometabolism. And this is based, the immunometabolism is based in the tight relationship between the immune and metabolic systems. And we have the ability to deal with pathogen and also the starvation resistance. Both characteristics make us adapt to the environmental change and survival. And it happens because both systems, metabolic and immune, share hormones, cytokines, signaling proteins, transcriptor factors, bioactive lipids, making us able to survive to the challenge of the environment. In this sense, it's well known that a normal body weight give a well-balanced inflammatory response. On the other side, when we have in a malnutrition and starvation, it is related with immunodeficiency. On the other side, we have obesity that is characterized by a low-grade chronic inflammation. And this history about inflammation during obesity development started almost 30 years ago when hot mildew and Spigma showed a higher expression and protein content of TNF-alpha in the adipose tissue. Since then, we know much more about this process, and we know that when adipocytes increase in size, we increase even in number, they started to show hypoxia in the adipose tissue. They start to have fibrosis, and they start to have the infiltration of a lot of kinds of immune cells to the adipose tissue. In this sense, we have an increase of pro-inflammatory cytokines that make a pro-inflammatory environment in the adipose tissue that can spread to other organs, and it is related with the development of a lot of kind of the disease. So we well know that in linear adipose tissue, we have a balance between anti-inflammatory and pro-inflammatory cells and cytokines, and it is related with the energy homeostasis. In the obese adipose tissue, we have an imbalance between pro-inflammatory and anti-inflammatory cells and cytokines that make this tissue start to have a dysfunction. And in this sense, adipose tissue expansion is related with a chronic low-grade inflammation that can trigger a lot of kind of disease. Our group have been studying not only the disease-related inflammation in the adipose tissue, but we started to argue whether this inflammation, this inflammatory response is related with some homeostatic function in regulating adipose tissue expansion. We know that we have in the adipocyte, we have a canonical way that control fat mobilization. It is related with activation of sympathetic systems that increase the lipolysis in the adipose tissue, releasing free fatty acids and glycerol. This is the regular pathway that control the mobilization of fat in the adipocyte. We started to ask if some inflammatory pathway, inflammatory cytokines or cells have a regulatory control in this mobilization. To doing so, we started to looking at the platelet activated factor receptor. This receptor is related with a lot of immune response, a lot of the production of a cluster of pro-inflammatory cytokines. And we used mice knockout for this receptor, the PAF knockout mice, to check whether in the absence of this inflammatory pathway, we have some change in the remodelation of the adipose tissue. We used a high carbohydrate diet to induce obesity in those mice. And as you can see here, wild type mice have higher adipose tissue index comparing to wild type. In PAF knockout mice, they also have a huge increase in the adipocyte size, adipocyte number, and it is related with the absence of this inflammatory signaling. As we can see here, the epididymal adipose tissue starts to become even bigger comparing to wild type mice fed the same high carbohydrate diet. We also checked whether it would be related with human obesity. And as you can see here, we have the expression of PAF receptor in the adipocyte and stroma vascular cells from obese patients. So they express this receptor in the adipose tissue, and this expression of this receptor is related with lower weight, lower BMI, and lower fat mass in obese patients, which means that those that have higher expression of Kupffer receptor are those that have less fat mass. Another pathway that we studied previously was the TNF factor, and it was a very simple way to show that when mice doesn't have the expression of receptor 1 of TNF, they also have a huge increase in adipose tissue, showing that for some reason having those inflammatory signaling active in the adipose tissue may control the adipose tissue expansion. Another pathway that we investigated was the IL-18, and we showed that knockout mice for IL-18 cytokine also show a higher adiposity index, higher adipocyte area. They develop glucose intolerance, showing another pathway, another cytokine pathway related with the control of adipose tissue expansion and metabolic control. We also worked with eosinophil cells, and specifically this work will be showed in detail in the afternoon or in the night, depending where you are, and by Marina. And in this paper, we showed that the absence of eosinophils in mice determine a higher adipose tissue expansion. They develop glucose intolerance, and patients that has higher markers of the presence of eosinophils in adipose tissue, including Singlet, IL-5 receptor, CCR3, are those that have better expression of glucose metabolism markers, including GLUT4, IRS1, and adipokine. It's not only our group that has been showing the control effect of inflammation modulating the adipose tissue expansion. We can see that other groups also have been showing that IL-18 is related with the adipose tissue remodeling. The absence of interleukin-1 receptor 1 also determine the adipose tissue, the expansion of the adipose tissue in young and old mice, and also the deficiency of IL-6 determine a mature onset of obesity. In this sense, we have been discussing the physiological role of immune system controlling the exacerbation of adipose tissue expansion. However, when it became a pathologic feature, as we can see in the adipose tissue, we have the loss of this control, and the mice start to become even fatter and develop a lot of impairment in the metabolic control that determine the occurrence of a cluster of other diseases, including type 2 diabetes, non-alcoholic fatty liver diseases, and so on. So if we have in mind that the a proper inflammatory response is important to control the exacerbation of the increase in the adipose tissue, it is also a good question to asking whether we need a proper inflammatory response to control the reduction in the adipose tissue when it is challenged during a fasting or a weight loss process. So the next question we are starting to try to answer is if inflammation plays a role in regulating maladaptive adipose tissue expansion. It is also instrumental in mediating fat mass reduction in the context of fasting or weight loss. Trying to answer this question, we have looked what we have previously published, and this is a remarkable paper published by Ferrand showing that a 24-hour fasted mice show a higher free fatty acid level as we expected, but they also have an infiltration of macrophages in the adipose tissue, and it is the first clue that we could see that during fasting, a prolonged fasting state, we have an infiltration of immune cells in the adipose tissue. And as TNF-alpha is an important cytokine, already showed be regulating the lipolysis in human adipocytes, we started to check whether these cytokines may be the core of this fat mobilization during fasting. What we did was to use in knockout mice for receptor 1, TNF receptor 1, to check whether when they are subjected to a 24-hour fasting, they would have a decrease in adipose tissue size and adipocyte number and so on. So we can see here that in wild type, subjected to a 24-hour fasting, they lose epididymal adipose tissue. They have lower, smaller adipocytes in epididymal and inguinal adipose tissue. However, when mice are knocked out for TNF receptor 1, they do not lose, as we expected, both adipocyte size, epididymal or inguinal adipose tissue. And it is related with a lower inflammatory response in this tissue, as we can see here in epididymal and even in inguinal adipose tissue. Following a 24-hour fasting, mice started to have higher level of IL-6, IL-18, in the inguinal IL-6, IL-10, and IL-18 in the adipose tissue. However, for those animals knockouts for TNF receptor 1, they do not increase those cytokines when challenged with a 24-hour fasting. And even when we increase the time of fasting, challenge mice for 48 hours fasting, we can see here that knockout mice are protected from the decrease in the epididymal and inguinal adipose tissue. They do not change. And the adipocyte size from those mice still are bigger compared to those that was not challenged with a fasting. And this is new data that we are still working on and trying to correlate the expression of TNF-alpha with fat mass, body weight loss in humans. These are from a group of obese patients that were subjected to a bariatric surgery and were followed for 12 months. In the orange, we can see the TNF expression correlated with BMI loss, and after 12 months of after bariatric surgery. And in yellow, we can see in the beginning, before the bariatric surgery. We cannot see any correlation at the beginning for body weight loss or fat loss or even for leptin. However, those patients that have higher expression of TNF after 12 months after bariatric surgery were those that have a higher body weight loss, higher fat loss and also lower leptin levels, circulating levels in after 12 months of bariatric surgery, showing that maybe the signaling of TNF is related with fat mobilization, fat loss, even in obese humans. So in this sense, we are working with the possibility that TNF is a key cytokine related to a proper adipose tissue reduction during fasting or obese treatment. However, we also would like to understand whether during a chronic low-grade inflammation seen in obesity, if this influence of inflammatory signals for remodeling the adipocytes size is also it's happened. So we argue whether how does the pre-existing state of a chronic low-grade inflammation in obesity state affect adipose tissue remodeling and fat loss during the weight reduction. One of the papers that we studied it was comparing the response of obesity versus lean mice following challenging for a 24-hour fasting. And here we can see that lean mice lose after 24-hour fasting, acute fasting, 63% of the epididymal adipose tissue. Around half of this is happening in obesity mice, showing that those mice are not able to lose as much as lean mice are when they are challenged to a 24-hour fasting. And it's very interesting to see as we expected, lean mice have higher expression of IL-6, TNF, IL-10 in epididymal adipose tissue, a feature that we cannot see when obese mice are challenged with a 24-hour fasting. So it means that despite they have basal highest level of cytokine in the adipose tissue, when they are challenged with a 24-hour fasting protocol, they do not increase those cytokines, as we can see as a response in Leon's 1. Interesting to see that the rolling and adhesion of leukocytes in the vessels of the adipose tissues are also increased in lean mice, are features that do not happen in obese mice. Despite they already have higher number of rolling and adhesion leukocytes in the adipose tissue, when they are challenged to a fasting, they do not respond as we seen in Leon's 1. So for both sides, when we need to reduction and we need to control expansion, inflammatory response seems to have a role. So in this sense, we have been arguing that although the inflammation plays a regulatory homeostatic role in the initial adipose tissue function, the chronic low-grade inflammation observed in obesity prevents adaptive adipose tissue remodeling during weight loss, a feature that we have been calling inflammatory inflexibility seen during obesity. As a proof of concept, we worked in a clinical trial with patients post-transplant of liver transplant. Now, we know that around 60 to 70% of patients that are subjected to liver transplant, they develop obesity. They need to take immunosuppressive medication at maintenance doses daily for the rest of their life to ensure the survival and function of the graft. So we choose to work with those patients as they are immunosuppressive patients, but they are still developing obesity in a higher prevalence. So what we did was to invite obese post-liver transplant patients that were obese and compare them with obese patients do not subjected to liver transplant, and propose to them to perform a dietary intervention trying to lose weight. And we followed those patients for 6 months. As you can see here, as we expected, liver transplanted patients have lower content, circulating content of a lot of cytokines evaluated, but no change in adiponectin, resistin, or leptin. And as they were subjected to a dietary treatment, we can see that the control ones, those obese patients, do not that didn't do the liver transplant, the regular obese one, let's say, they were able to lose weight, to lose fat mass, a feature that we cannot see in patients that were transplanted before. So they didn't respond to the dietary treatment. And it was very interesting to see that as much as they have tacrolimus in their serums, they have lower body weight loss. So the residual tacrolimus concentration in serum of those patients determine lower response to the dietary treatment aiming to lose body weight. So in this sense, we are still working in some pathway that may be related and add some information about the whole regulation of the mobilization of the adipose tissue. So we are including more insights about the inflammation, controlling and acting as a partner of the canonical pathway to mobilize fat in the adipocyte. And we are also working, trying to understand whether during obese development we lose this inflammatory flexibility that make trigger resistance to lose fat when we have a chronic low-grade inflammation. And that is the part of the team. I don't have photos of all of them, but here we have important people that we have been working together for a long time and in a happy way to develop science as we, and same time that we enjoy doing it. So I would like to thank my group, Immunometabolism group, nutriomics, immunopharmacology in a special way. I also would like to thank all the collaborators that are here and developing new projects with us and enable us to be here exactly right now discussing insightful and interesting ideas in the field of immunometabolism. Thank you so much. And I will stop to sharing here to see you now. And we have some times for questions. And I am the chair and I am the speaker, so I have to understand here. And we have the next speakers. But I think we can discuss if someone has some question about what I have just presented. Please feel free.
Maybe Dalian, I can start a question. Thanks a lot. I think this concept of inflammatory inflexibility, immune inflexibility or inflammatory inflexibility is really, really interesting. And I guess one related question is that, you know, even not in severe obesity, but we all know that with patients there is weight loss, weight regain, and, you know, this so-called yo-yo
Yes.
characteristic. Do you think that this could be also a factor, while you don't necessarily start with lots of inflammation in the adipose tissue, but that the yo-yo phenotype could really induce per se this
Yes.
inflammatory inflexibility? And do you think we in terms of experiment, we could test that also in human?
Yes, it's a very, I have two things that I have been thinking deeply in this is the yo-yo feature of a majority of the patient. And I think that they lose the ability to proper mobilize because they became resistant to respond to the homeostatic signaling, including cytokines. This is one way. And we have Laura that is working in this possibility, working in a yo-yo protocol to check whether as much as they became formerly obese mice, they became more resistant to this loop of inflammation response, like let's say. And another interesting thought is how can we achieve so higher weight, body weight and body fat without dying? And I'm thinking this is one thing I would like to have time with you after to discuss, because I think that those severe obese patients are those that do not inflame properly and do not have the control of the obesity development. In this case, they can increase their fat mass without developing shortly disease. I'm not saying that they will not develop disease. I'm not saying that, but I'm saying that we have a lot of severe obese patients that do not have change in their glucose level, in lipid lipoprotein. So maybe they are more resistant to this.
No, I agree. Actually, I think it's really interesting, and I agree with you. We did that in the past with you know, Prader-Willi patient or you know, people with genetic form of obesity where you have really fat mass inflation and basically for a long time no abnormality in glucose metabolism. And so what we observe is decreased inflammation in the adipose tissue as well as yes, the decreased inflammation and basically extracellular matrix is not so altered, but that's probably also something we should consider coming back to that and doing it well, because we haven't been so far into that. But yes, I agree with you. But really, this concept, what you showed, it could be really interesting if you induce so kind of a yo-yo and repeat after the yo-yo what you showed about 24 hour
4 hour fasting
or 48 hours fasting. So maybe after 2 and 3, even if the start is not severe obesity like mild obesity with eventually mild inflammation, it could be really interesting to have a mild phenotype and whether you aggravate this dysfunction with the yo-yo. Yeah.
Yes, I agree with you that we have to include a challenge for those mice, because we know that when they lose weight, they will have lower inflammation in the adipose tissue. But the question is whether when they are challenged. So we need to perform the 24-hour fasting after all the protocol. And this is what we are intending to do with Laura. We can discuss. But thank you so much, Karine.
Thanks to you. Yeah, very nice presentation.
It's so nice. And now I have to present you for our audience, no? And it's a great pleasure having Karine Clément here. She is a professor in the head of nutriomic research team at the renowned French National Institute of Health and Medical Research, and also serve as a professor of nutrition. Her work is pioneering and groundbreaking in the understanding of obesity and adipose tissue dysfunction, and she is internationally recognized for interacting study on genomic, metabolomic, and the influence of gut microbiota on the pathogenesis of metabolic disease. So please join us in extending our warm welcome to Professor Karine Clément. Thank you so much, Karine.
Yeah, thanks a lot, Adalienne, for this very kind presentation. So actually, I changed a little bit my title because I saw you had this introduction on adipose tissue and inflammation. So, but it will be more or less the same as you mentioned in the title you propose. So you'll see, but I tried to link some information between gut microbiome and adipose tissue, actually. So I will, okay, so can you see the slide well? Is that okay?
Yes,
Yeah,
yes, it's okay.
Yeah, perfect. So I'm going to speak about a part of the work we are doing in gut microbiome in people with obesity, and I will indeed focus on the crosstalk also with adipose tissue. Okay, so yes, a little bit of numbers. I just focus on the numbers. You have the numbers of inhabitants in Europe, of humans on Earth, and if you see the number of zeros for bacteria per intestine, so you can see that it's really in trillion. And also some species have been identified, about 4,000 or so species in human intestine. And of course, I won't come back on number of people with obesity in Europe and overall in the world. You are all aware about that. And so what we learn with time is the fact that the gut microbiome exerts key function in our biology, and I will indeed show some example here. So when we think of the gut microbiome, I can say that we learned a lot in the last 20 years, where there is really an interplay between environmental lifestyle factors, of course, subject biology and the biology of the intestine, of the gut, and the microbiome. So I really like these slides, and it was a review performed by colleagues in Belgium, where basically what you get here is the microbiome. So you have different metrics, like the diversity, composition, function, and the capacity to produce gas. And something also very important, and by the way, that echoes what Adalienne you showed, basically is the capacity to restore and have a resilience function. Basically, you have an alteration, and just after the alteration, you come back to a proper functional microbiome, and to some extent that's a little bit like that for adipose tissue, where we all think that the tissue should keep its plasticity. Then you have the side of the gut, and we spoke about inflammation, and this is also the case for inflammatory markers, and there are some markers like calprotectin, for example, you can measure in fecal samples. Obviously pH, that can be different along the gut, and the bacteria produce many metabolites, and there are key metabolites. There are many of them. I'll come back on that: short-chain fatty acid, bile acids, tryptophan derivatives. So what we learned in the context of obesity is the fact that many aspects of this function of the microbiome and the gut are altered in people with obesity. We speak about change in diversity, in composition, and in function, as well as modification of myriads of metabolites. Inflammatory markers, so Adalienne, basically you spoke about that in the blood, so there is really systemic inflammation. And this slide is to show you the side actually of inflammation if you look at jejunal. So it's a paper we published already 10 years ago, where we had access during endoscopy to jejunum, upper jejunum fragment. And it's basically at the duodenojejunal area, where we were able not only to look at the intestine and its function, so we measured, as you can see here, the velocity of the intestine, but also immune cells. And to make it short, basically what you can see here is that in people, so you have metabolically healthy subjects here, people with obesity and diabetes, you have a modification when you compare to lean subject, modification in accumulation of immune cells. This is an example here for lymphocytes. And what we showed also is that some of these lymphocytes, we were able to stain the lymphocytes along the epithelial barrier, so between epithelial cells. And again, if you have a marker here for macrophages, for example, you can see the augmentation of macrophages accumulation, and you can see it here. It's always true when we look at metabolically healthy subjects, so early unhealthy subject, unhealthy subject, basically. So there is early augmentation of this type of cells, and it's not the same for all the cells, and you have an example here for mast cells with tryptase and chymase, and it was not different. So in this work, we show that basically T cell density were correlated also with the level of corpulence, liver disease, and lipid metabolism. And with some in vitro experiment, I won't detail too much, but we showed that the media, the cytokines, were involved in the impairment of intestine function, and notably insulin resistance. Okay, so we're speaking of obesity. So, and I like very much these slides here, which is you can see early in the 2000, where the different actors are known. So behavior, genetic, epigenetic, of course, lifestyle. So that's people due to individuals, but also we all know that's a systemic disease, and there is a global collective responsibility, especially based on environmental changes. And now we have some new actors, and this includes the gut microbiome that could contribute to perturb interorgan function. Okay, so Adalian spoke about that. Basically, we have a tissue, and in the context of obesity, its function, its structure and function is altered. And within also our consortium, so Geneviève Marcelin has worked a lot on extracellular matrix and the remodeling, and also the reason for accumulation of collagen here in this tissue. And just a review from also Simon in the lab, recapitulating the different modification of adipose tissue, which include indeed hypoxia, catecholamine resistance, hyperlipolysis. So basically, what we could say is that the proper fate of the adipose function is deeply altered in the context of obesity. So what I'm going to speak about here is a result of long-term work, especially in the context of this project called Metacardis. And this was a European project that I coordinated in Europe. And what you can see, there were 14 partners in 6 European countries. And the idea was really to study the impact of qualitative and quantitative change in the gut microbiome on the pathogenesis of cardiometabolic disease and associated comorbidities. And it was really a large group with transdisciplinary expertise, with many contributors, people involved in metabolism, microbiology, immunology, omics, data analysis, and so on, as well as patient association. So I give you an idea of how the project was organized. So basically, we constituted a large European cohort, more than 2,000 subjects. We had lots of work in performing core phenotyping, including lifestyle and nutrition related data. We performed molecular phenotype and did an analysis, and then developed pipeline to do multi-omics analysis. And obviously, when you do this kind of things in human population, so you generate hypotheses. And really, the idea was also to have some approach in preclinical model to confirm and perform in vitro and in vivo models to confirm and explore some of the identified targets. And it could be, for example, a new bacteria or a metabolite. And for maybe younger people listening this talk, what I want to mention is the fact that when you do this type of approach, you can see here, you have a different level of information, obviously, clinical, lifestyle, nutrition-related factors. We also measure microbiome cell density, metabolites, fecal metagenomics, urine metabolomics. And if we want to get a complete set of factors, obviously, this reduces also the number of individuals, but nevertheless, it was not too bad. And here are the first main message we get with this work. And this is a summary of some of the work, and then I will do some focus. So first of all, what was really interesting is that in this consortium and the patient population we had, it was a cohort with patients at different stages of their cardiometabolic disease. So basically, we recruited patients with no disease, healthy people with overweight and obesity, but with no complication, and people with cardiometabolic disease. And when you do that, obviously, because really the microbiome is at the interplay between nutrition, lifestyle, and our biology, these people are taking, obviously, a medication, especially if they have a cardiovascular disease. So first of all, what was really important was to develop a pipeline for complex data analysis, and this was done by colleagues in Germany. And it was very important to see that some signal in the gut microbiome is due actually to medication. And so it's important when you do that to disentangle and to have a proper signal for disease, keeping in mind that you need to take account polypharmacy. Another very important message here, and I'll come back on that, is that we also observe, especially in the context of obesity, specific bacterial signature really associated with obesity. And related to that, when we look at the progression of the disease among the different groups, we observe that this early bacterial signature were already present in people with overweight and obesity, I mean before a more severe state of the disease. And then we had some focus in this work about some metabolites, and among the metabolites, there are some functional alterations. We were able to observe, for example, that there is a deficit in some B vitamin in severe obesity. And we had also some focus on some targeted metabolites. And what we are doing also, and basically the consortium is still alive, and we are doing also some work on obesity-related complication, especially, as you can see here, chronic kidney disease. Okay, so let's come back on overweight and obesity. So this was basically an historical exploration here, where we explored one of the phenotype, which is very important, which is microbiome richness. And what you can get here is that because we get metagenomic data, we are able to count the number of genes. And then with the number of genes, you have an idea of the microbiome richness. So basically, you have individuals with higher richness and individuals with obesity, but lower richness. And by the way, you can see here, people with severe obesity going for bariatric surgery. You have about more than 70% of this person being in the low richness group. And what you can get here is that we had a negative association between gene richness and the accumulation of body fat measured by DEXA here. So this gives you an idea of the interplay between, of course, the microbiome, especially the richness, and the adipose tissue. Then second, when we carefully look at the bacteria, we have, especially in the low richness group, enrichment of bacteria with pro-inflammatory properties. And of course, with clinician, you can separate and explore those individuals with higher richness and those with lower richness. And we showed that a low gene count, meaning lower richness, associate with a series of cardiometabolic risks, such as dyslipidemia, adiposity, insulin resistance, and low-grade inflammation in the adipose tissue. So I mentioned some specific bacterial constellation, and you have an idea here where you have a principal component analysis. And basically, you have a group of individuals where you can see some specific bacterial regroupment. And we were very interested here in this so-called BAC2 constellation, which associate with some bacterial group, but also, and this is probably more important, lower proportion of some beneficial bacteria, which are Akkermansia muciniphila or Fecalobacterium or Methanobrevibacter proportional abundance. And so this bacteria, the amount of this bacteria is decreased, and this led in this constellation to a decrease capacity to produce butyrate. And what we showed here in this group is on the other hand, there was highest capacity to produce trimethylamine, for example, and we show some link with complication. And this is Carlota Dao, who was a postdoc at that time in our lab. And what Carlota did was to combine richness to microbiome richness to the abundance of this interesting bacteria called Akkermansia muciniphila. And to make this long story short, what you get here, if you look at patient phenotype, those with a better metabolic profile with lower android fat, decreased glucose and triglyceride, were those with higher gene richness and higher abundance of Akkermansia muciniphila. But you can get here that numbers are quite small because it was performed in people with overweight and obesity. So, but in Metacardis, we had higher information. Again, you see this principal component analysis, where you retrieve here in red a group of individuals with BAC2, and you can see this positive association between fat mass, triglyceride, body mass index, and BAC2. And what was really interesting, again, is that this BAC2 bacteroides 2 phenotype was associated with lower microbiome gene richness, either counting the genes or even counting the amount of bacterial cells to measure microbiome density. And what you get here, if you look at people with increased severity of obesity, you can see body mass index from 20 to 70 here. You can see that there is increased abundance of BAC2 with the severity of obesity. And it was replicated in another population cohort. And we had also association between BAC2 and low-grade inflammation measured in the blood. And just a little for Adalien also, what we discussed when you came, we discussed about the gut microbiome, but we could try to build these different type of constellation, like enterotype, and look at the link with low-grade inflammation measured either in adipose tissue or in the blood. Okay, so then I spoke about some specific bacteria, gut microbiome, but obviously this bacterial group are also involved in the production of metabolites. And during the consortium, the group of Frederick Backhead has identified a metabolite called imidazole propionate. And he did this very nice paper showing that basically this metabolite can be produced by the bacteria, and the bacteria use histidine, and the bacteria have equipment in enzyme that produce imidazole propionate, and the group of Thierry Huby showed the relationship with impaired glucose metabolism, with notably a phenotype on the liver, showing decreased AKT phosphorylation in liver and a decreased expression of ISR1, for example. So then what we did was to look in the in the Metacardis cohort, where what you see here is that when you are in the highest quartile of imidazole propionate measured in the blood, you have increased risk of type 2 diabetes. And here it's just a matter of different model and different adjustments in the model. So at least you have an odds ratio over 2. And we look also the links with comorbidity, and in people with obesity, there is a strong risk with heart failure. And we show that those with the highest level of imidazole propionate have more cardiovascular events. So there is probably a link with complications such as heart failure. But again, I spoke and I told you about this BAC2 enterotype, and what we showed is that when we look at the constellation, the different group of metagenomic clusters, those in the BAC2 enterotype had increased blood level of imidazole propionate. You can see that here. And we look at the abundance of the genes in the bacteria that are able to produce imidazole propionate. Then, obviously, one of the questions was, what about food and what people are eating? And since histidine is really a precursor of the synthesis of imidazole propionate, so we measured histidine in the food, and you can see here by quartile of histidine quartile of imidazole propionate, you see that the level of histidine intake was the same. So it's probably more a pattern of food intake. And again, what you get here is that we had this scores, diet quality score here, and we have a negative association between the score of quality of food, or the DASH score, or diversity score, or Mediterranean diet score, that were negatively associated with the blood level of imidazole propionate. So this gives you an idea of the interplay between food patterns, microbiome, some targeted metabolites, and here we spoke of the metabolic risk, such as, for example, impaired glucose metabolism. So this comes to indeed this interplay I just mentioned between lifestyle, microbiome, the contribution of metabolite, and a series of phenotypes we just mentioned: metabolic phenotype, inflammatory phenotype, immune, and so on. And this is what we're exploring now, even combining some of the lifestyle factors, including physical activity, or even smoke, or even dietary patterns. So a way, and then if you look at not only the diversity score, but this lifestyle score, meaning that a higher score is related to a better lifestyle, you can see the association between a higher lifestyle and increased gene richness in the microbiome. Again, here on the left, you recognize the Bacteroides 2, and there is an association between a low lifestyle score and this enterotype called, again, BAC2. So we did a lot of studies on that, and with some targeted metabolites that are enriched in the high lifestyle score, or on the other hand, diminished in the low lifestyle score. So now we are better exploring the interplay with especially the immune system, or low-grade inflammation. I'm going to skip that one. Just a really brief notion is that we explore causal inference in this to really explore the different direction between the lifestyle, metagenome, and phenotype. And you can obviously use also the metabolomics, blood metabolomics, as a precise human phenotype. Okay, so I just spoke about some metabolites, but and you see, I like very much this slide here. It's quite complex, but what you get, you can follow basically the boxes and the color of the boxes. And if we focus on adipose tissue, basically you can see you have the gray here, the yellow, and the red with what inflammation, and the relationship with some of these metabolites. Here in gray, short-chain fatty acids, or in yellow, you have bile acids, or again, in red, you have a link with some toxins, for example. So it's interesting to see the relationship with the complication. Okay, just to finish, to modify the gut microbiome, you can do dietary intervention, of course, bariatric surgery, and now medication. And again, this is a older study here where we perform a dietary intervention with caloric restriction, and this diet was particular because it was a moderately restrictive diet, but enriched in fibers and protein. And the main message here is that those individuals with low gene count, so low gene richness before the intervention, had a 30% increase in gene richness. And you can see it was quite short, 6 weeks. And you can follow after 12 weeks, it remained low. And those with high gene count before the intervention remain with high gene count. And it even seems that there is a small decrease here. And we found in this study a negative association between variation in microbiome richness and macrophage accumulation and low-grade inflammation in this subject. Maybe I will skip that one because I'm seeing that the time is flying. Again, if we look at bariatric surgery, you can see that people with gastroplasty, gastric ring, increase richness after bariatric surgery, and here even after gastric bypass, but not enough to reach the mean. Focus on Bacteroides B2, I mentioned before. Look, after bariatric surgery, actually, this disappeared. I'm going to skip that one. Just here to mention that what was really interesting in this work is the fact that we had increased also abundance of some bacteria such as Akkermansia mucinifila, but we were not able to see association between this change and metabolic improvement in people with bariatric surgery. While we show some interesting association. For example, here, the blue one is its increase after bariatric surgery is associated with the diminution of adiposity marker, or the other way around, diminution of some bacterial group associate concomitantly with reduced body fat mass and reduction of body fat. I'm going to skip here. Just another quick example here. This is a study we are doing with Signe Torekov in Copenhagen, and she did a very interesting work she published in the New England Journal of Medicine in 2021, and this was with GLP-1 analog, and it's a weight stabilization study. So basically, they did a weight loss phase with a very low calorie diet, and then the patients were randomized to placebo, exercise alone, liraglutide, and the combination exercise and liraglutide. And what we observe is that we had access to the fecal sample, and the study was done by Roya Alili and Eugénie Belda. And you have, again, in this population, with people with overweight and obesity, we have, again, people with low gene count and high gene count. It was amazing to see that during the very low calorie diet phase, there was, exactly as we've seen in the previous study called the microbe study, those with high gene count before, during the VLCD, there was no change, while there was an improvement for those with low gene count starting during the VLCD. So what we are doing now is that we are exploring the change in the microbiome in the different arm of treatment. And you can see we can do clusters of weight stabilization, weight loss, or weight regain. So I won't detail too much. It's just an example of studies we are doing. So just to finish, basically, you know, as clinicians, so we have patients with many types of information about their characteristics, their phenotype, their comorbidities, and so on. So this question is the use or not regarding the information of the gut microbiome. We know that if we do an intervention, we would modulate the gut microbiota, and there are different ways to modulate it. Weight loss, use of prebiotics, etc. We know this has an effect on the microbiome. This change many factors: ecosystem diversity, change of abundance of microbiome, beneficial microbes or metabolites. And in patients, we expect an effect. But what we need is really this relationship here. And for most of the case, the question is which part of this modification in the microbiome could explain some of the beneficial effect of some diet in the patient. And that's really something we need to explore more. Okay, so I show you a lot of association, and you know that correlation doesn't mean causation. And just to finish on that, again, I won't detail too much, but you can see the complexity here. If we think, for example, of adipose tissue, you have an example here, potential for beiging, adipogenesis, lipogenesis, and so on. Many of these receptors are expressed on the adipose tissue, and some products are coming from the gut microbiome. So there's a lot to explore here. Also regarding brown adipose tissue, and especially regarding mitochondria and coupling. So I hope in the future we can do this kind of approach, linking microbiome changes to a mechanism, especially focusing on the adipose tissue. Okay, so just to finish, I showed you an example of these large-scale studies we can do, and one of the main messages is that there is altered composition and function in the microbiome in people with obesity. I spoke about some specific phenotype of the bacteria, like Bacteroides 2. I spoke about some metabolites production. I didn't speak too much about vitamin, but that's also something interesting. And there are bidirectional interaction that needs better investigation. Regarding this BAC2, what we observe is that it's associated with severe disease severity and low-grade inflammation. And some of these changes are partially corrected by different type of intervention, but again, this needs to be explored better. And probably medication are not only confounders, but then the question is whether basically microbiome could impact the response to medication. And there is really a need to address mechanistic aspect. And I'm going to stop here. So you have, of course, the group and people here in the at the international level. Okay, thank you. I was maybe a little bit long.
No, it was amazing, Karine. Thank you so much for your talk.
Thank you.
And it's always incredible see how much you have been working in different fields, and considering microbiota, it is fantastic to see your trajectory, your journey through this field. So thank you so
Adaliane.
much.
And just I wanted to show that because indeed in the clinical protocol we discussed when you came,
Yes.
yeah, that's just you get an idea of this kind of pipeline and data integration we can do now in the lab.
Yes, yes, and I really hope soon being working with you analyzing microbiota from our clinical trial that we are running here. So it was amazing to have a deeper idea about what we can do together. And we have some questions for you.
Okay, great.
And here in this platform, if they are not speakers, they cannot do this. So I will read
Okay, no
all
problem.
of the questions. We have the participant, Satu Pekala, she make not a question but a comment saying that Methanobrevibacter
Yeah.
is many times bad and associated with obesity, insulin resistance, and type 2 diabetes. It's just a commentary. I don't know if you would like to comment on this.
Well, it's sometimes difficult to focus on one bacterial group because it's really a matter of ecosystems. I think it
Yes.
depends. And I can take also the example of Akkermansia mucinifila. And that's something I did not mention here, but, it has been seen as a beneficial bacteria and there are many data on that. But what is known also is that the gut barrier is altered. For
Yes.
example, the mucus layer is very low, very diminished. Then in that case, Akkermansia, if Akkermansia approach the epithelium layer, you can have inflammation actually. So it really depends. Sometimes it's difficult to isolate one bacterial group. I guess we need to keep in mind it probably depends on the other ecosystem, whether the microbiome is diverse or not, and so on.
Yes, for sure. Well, Ana Flávia Martins, it's a comment, not a question. Perfect. I work with metabolic programming, microbiota, and insulin resistant, and we are finding similar results.
Great.
And great, yes. And another is from Marina Oliveira. She said the microbiota is diverse among people with obesity. How would be the most effective way to change microbiota in obesity to reduce the presence of bad population? Diet, medication, probiotic. Are you thinking about different intervention according to these different profiles in each group of patients with obesity?
Yeah, no, that's a very good question because as you said, so first, what would be really great to think of in terms of intervention would be also to stratify by baseline microbiome. And this is why I wanted to show you, based on the diversity, low diversity, high diversity, the response can be different. Including the response to different type of as you mentioned, fibers, probiotic, and so on. But in terms of immunometabolism, indeed, I mean, we are starting some project now to go back to so-called anti-inflammatory diet, such as Mediterranean, whatever, to study more deeper the interaction with low-grade inflammation. So a point is also the capacity, and something which is not known is related to resilience. So when you have a switch in bacterial diversity and richness, are we really able to restore it? And what is the point, really the point of no return? And this is what we'd like to study more, because maybe sometimes you have diminished richness, you can, by fibers, you can improve some beneficial bacteria and so on. But maybe at some stage, when it's so altered, you need other means. So maybe bacterial constellation combined with prebiotics. People are doing some phage approach also now. So yeah, that's this kind of things. But we need proper intervention to explore that in human. Yeah.
Yes, it's a good, big challenge.
Yeah, sure.
And the importance, we can see here the importance for individualized treatment for each characteristics of the patient.
And especially in some so-called precision nutrition, in some subgroups, which type of intervention could be interesting. And also, which part of the microbiome change explains the improvement. But obviously, it's only a part. And we need to explain that, we need to identify that better.
Yes, for sure. Well, we have a lot of comments. I will go further. We have Alina. Alina is asking, what are your thoughts on the use of pro-resolving molecules in the gut microbiome and the cardiometabolic disease in the context of obesity? Is it a possibility?
Good point. I don't know.
Yes,
No,
I don't have an idea.
Yeah. No,
And
yeah.
I know
I
why
mean,
Arina is asking for it,
Yeah.
because we have a project.
Yeah.
Alana is
Yeah.
the undergraduate student that are supposed to go to your lab
Okay,
as a
nice.
visitor.
I'll
And
be happy to
she,
discuss about that.
yes,
No, I mean, the point is, but the point is very interesting. I mean, it's a matter of resolution of inflammation and how this could act eventually. Eventually some molecule could act, whether if you
yes,
change. And this is why we need to keep in mind it's really bidirectional interaction. So you can
yes,
act on the host by, for example, anti-inflammatory molecule, or why not resolving whatever. And this
yes,
could also impact on the other way. And that's what we are doing to explore with causal inference. We are
yes.
exploring that. Yeah.
This is a topic I would like to discuss with you, because we are developing here bacteria that express an annexin 1
Okay.
A1, and we are trying to treat mice with those bacteria that overexpress this, and it would
Interesting.
be very interesting because it has a pro-resolution action, but we don't know a lot, but
Yeah.
Alina is supposed to work with this, and it's really interesting to check this in the context of microbiota. So the other is we have more, but I will just finish with the last one, okay? Otherwise,
Okay.
our
Yeah,
time goes
Thierry is
so
waiting.
exactly.
Yeah.
The last one, I would like to understand better how the BAC2 enterotype is defined in practical terms. Could you elaborate on its main microbial characteristics and how reliable it can be identified in different data sets?
It's an excellent question. So I think it would need a long development. Just keep in mind something important is when you do this enterotyping, you do basically a clustering by different methods. So you look at enrichment of some bacterial group that enable the clustering of the people. And so we have a project with our colleague Jean-Daniel, who is doing lots of study on the microbiome, exactly on that, how stable it is depending on the population. Because obviously, if you have, let's say, you take metacarditis population, 2,000 subjects, then you add another population on that, and you need to redo each time. So some of the phenotype of the enterotype are not stable. And some people in BAC2, if you change the population, would switch, for example, to BAC1. So this is what we are exploring. So it's not like, because people at the start say it's like a blood, blood group. It's not the
Yes.
case. You have a continuum. And then this is why when we explored that, so it's not only an enrichment of some bacteroides group, it's also a loss of some group. This is why I mentioned the decrease of Akkermansia, the decrease of Faecalibacterium proteinsis, and also at the functional level, there are functional alterations. For example, in the capacity to produce short-chain fatty acid, such as butyrate. So this constellation of group, yes, first, the question is really very important. It might be not stable depending on the population and depending where you position also the person in a group.
Okay.
And we explore that. Yeah.
Okay. Thank you
Thanks a lot.
for your great talk, to be here with us. It's a really pleasure that finally we were able to have our scientific meeting. And I'm very happy and proud that we could really perform it. Thank you, Karine, Clément. I hope see you soon. Okay, so I'm sorry, Thierry, but
No
it's
problem.
special for us to discuss as well, and I'm very happy to having you here. And I will introduce you so you can start, okay? So Thierry is a dedicated research at INSERM, the French National Institute of Health and Medical Research, contributing significantly to the field of metabolism and cellular function. His important work is focused heavily on lipid metabolism, cholesterol transport, and lipoprotein function, providing crucial insights into mechanisms that regulate the interplay between lipids and metabolic health. So Thierry, thank you so much for being here with us. It's your time.
Okay, thank you. So I will share my presentation. Oh, okay. Okay, why is it? Okay, if I do that. Okay, I have to change my parameter. Okay, here, all right. Ah, I'm sorry, I will have to just exit the meeting and connect back, okay?
Okay, no problem.
My
No
computer
problem.
is not
Take
allowing
your time.
for sharing otherwise.
Okay.
I'm sorry. Okay. 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 Autoriser. Can you see the presentation?
Yes, it's
Okay.
working. I guess you can, yes,
All right.
widescreen.
Okay.
Everything is good.
Alrighty. Okay, what am I doing here? I lost it. All right. Okay, so thank you, Adalien, thank you, Geneviève, for the invitation and to present some of the data and some of a potential immunometabolic link we have worked on and published last year, looking at the role of Kupffer cells and hypercholesterolemia. Okay, so this work, okay, how this is working. Okay, I don't know how to. Do you hear me?
Yes.
Okay, I'm sorry. I cannot change my slides and what is happening? I'm sorry. Okay, sorry for that. Okay, so this story started looking at some published results showing an inverse relationship between the number of macrophages and cholesterolemia. So these studies were performed using transgenic mice, as those models that did not really look at the one specific tissue macrophage population and what was causing this inverse correlation. So, as you know, macrophages are present everywhere in every tissue, every organ. So we decided to focus on one specific organ, which is the liver, because this is a really key organ in lipoprotein cholesterol metabolism, of course. And it also contains the largest macrophage population, which are Kupffer cells. So livers that are constituted, as you know, as repeated modules, and those modules, they contain a central vein connected to hepatic arteries and portal veins and through a sinusoid. And in these sinusoids, as you can see here, Kupffer cells are residing. These Kupffer cells are really strongly attached to the endothelial cells and communicate with surrounding cells, which are going to be those endothelial cells, hepatic stellate cells, and also hepatocytes. So they have one arm into the parenchyme, and they have the other arm into the blood flow. And this is really clearly illustrated with these two photon microscopy live imaging here, where you have these Kupffer cells here in red, in close contact with the blood, where you can see all the flow of erythrocyte going through. So these Kupffer cells, they interact with their surrounding cellular cell type, which are those liver sinusoidal endothelial cells, those hepatic stellate cells and hepatocytes, from which they receive information, signaling that will be very important for their identity, but also for their maintenance, for their capacity to survive and proliferate. And one example is this cytokine CSF1 that signals through this CSF1 receptor, and which is very important for Kupffer cell, but also macrophage maintenance and proliferation. On the other hand, Kupffer cell will also produce growth factor that will also help those surrounding cells to maintain. And this is, for example, the case for PDGF and hepatic stellate cells. So Kupffer cell function have been studied for some time now, and of course they play a very important role in immune surveillance. There have been many papers working on that subject. They also play a very important role in removing debris or modified cells, dying cells, such as erythrocytes or platelets, which have been modified. And they also play, and this is a more recent study that suggests that Kupffer cell could also have a role in the metabolism of lipid and carbohydrate. So this is why we focus our attention on those Kupffer cells. And to study the relationship between these Kupffer cells and cholesterolemia, we use a very well-known mouse model, which is the LDL receptor knockout mice. Can I remove this? Okay. This LDL receptor knockout mice, these mice, if you put them under either chow high cholesterol diet, which is the chow diet containing cholesterol, for 1% cholesterol, they will develop a buildup of cholesterol into the plasma, like hypercholesterolemia, reaching about 1,000 milligrams per deciliter. So we have studied the leukocyte population. We have studied the leukocyte population in the livers of those animals fed either the chow or the high cholesterol diet. And this has been done by flow cytometry. And here you have a 2D projection of those leukocyte population, each color representing one cell type, for example, the B cell here in gray. And you have here the Kupffer cell here in orange with the expression of basic-4, which is a typical marker of those Kupffer cells. Now, if you split the two conditions, you have the chow condition and the high cholesterol condition here, everything is pretty much the same for all liver leukocyte population, except for the Kupffer cell, where you can see there is a big difference between the two conditions, which was showing us that this Kupffer cell pool was markedly perturbed by hypercholesterolemia induction. So we went further to analyze the Kupffer cells, which by flow cytometry, those Kupffer cells have CLEC2 positive cells, and they express a number of very specific markers, CLEC4F, basic-4, TIMD4, and those markers are specific of those embryo-derived Kupffer cells, and they were both present on the chow and high cholesterol condition. We then evaluated the density of those Kupffer cells between the two conditions and observed that under hypercholesterolemic condition, those Kupffer cell counts were markedly increased. These results were confirmed by immunofluorescent microscopy with Kupffer cells identified with the CLEC4F marker. So here in red, the green color is for e-cadering that delimit the portal regions. And as you can see, between the two conditions, we could also evidence clearly that Kupffer cell counts were increased under hypercholesterolemic condition. So we went on to sort by fax those Kupffer cells and study their transcriptomic by doing RNA sequencing. And looking at the gene upregulated in the high cholesterolemic condition, we can easily identify a number of markers for proliferative cells, and notably this Ki67 marker that we use by flow cytometry to confirm that those Kupffer cells were indeed proliferating. So meaning that this pool expansion was due to Kupffer cells proliferation. To search for signaling that could trigger this proliferation, we turn to this CSF1 cytokine and look at this abundance of the mRNA in the liver during hypercholesterolemic feeding from day 0 to day 4, and you have an increase in these cytokines in the livers of the animals. We try to identify which cell were producing this CSF1 in the livers. So we sorted stellate cells and liver sinusoidal endothelial cells to look at the presence of CSF1 in those cell type, and we could see that hepatic stellate cells were the cell type that was producing CSF1 in a larger quantity. We also look at the density of those hepatic stellate cells using desmin as a marker. And we could also see that those hepatic stellate cells were also expanding under hypercholesteremic conditions. So basically, these cells, which are in close contact with Kupffer cells, expand and produce more CSF1, which suggested that this was a signal for Kupffer cell proliferation. To truly demonstrate that proliferation was triggered by this CSF1 receptor pathway, we use specific inhibitors for CSF1 receptor, and we fed the mice either with a high cholesterol diet and high cholesterol diet added with a CSF1 receptor inhibitor. And by doing so, we could see that even under high cholesterol condition, Kupffer cells were no longer proliferating, and we did not see the expansion of the pool. So truly demonstrating that this inhibition of the CSF1 receptor was responsible for proliferation and UMKC pool expansion. We further analyzed our RNA sequencing data on unsorted Kupffer cells and could see that they were enriched in a foamy macrophage gene signature that was suggesting to us that those macrophages were loading lipids. And indeed, this was true as we perform flow cytometry analysis using the BodyP, which is specific for staining lipids in cells. And we could easily demonstrate there was a very strong enrichment in lipid staining in Kupffer cells under the hypercholesteremic condition. And we went further doing unsorted Kupffer cells a lipidomic analysis, and we could clearly demonstrate an enrichment in free cholesterol and cholesterol esters in those cells without major changes in triglyceride in those Kupfers. We went on to further visualize these lipids in those Kupffer cells. We used 2-photon microscopy using CARS, which allowed us to see the lipids represented here as the white patches here under the high cholesterol condition, big white patches, which are lipid droplets in hepatocytes. And when we focus on Kupffer cells here, seen with the TIM-D4 marker, we can see small lipid droplets present in those Kupffer cells. So looking at the lipidomic data, so under the high cholesterol condition, we can see that there are a twofold increase in total cholesterol in the livers of those mice, which is well known. They accumulate cholesterol in their livers. And looking at the amount of cholesterol that accumulates in the whole Kupffer cell pool, this was no longer a twofold, but like more than a 30-fold increase in total cholesterol accumulation in the cells. However, this amount of total cholesterol is only 1 out of 400 of that present in the total liver cholesterol, meaning that these Kupffer cells accumulate large amount of cholesterol. However, this amount is only a small part of all cholesterol accumulated in the liver, which is stored mainly in the hepatocytes. To try to understand how those Kupffer cells were acquiring lipids and cholesterol, we look at the RNA sequencing data, focusing on scavenger receptor, as we know that macrophages are equipped with a large number of scavenger receptor that help recognizing modified lipoprotein, notably, and one specific element is CD36, which was clearly upregulated in the high cholesterol condition in Kupffer cells. And so we tried to demonstrate whether this CD36 scavenger receptor was involved in lipid uptake. So we took the LDL receptor knockout mice, fed them with high cholesterol diet, and injected them with an anti-CD36 neutralizing antibody. And this resulted in a decreased amount of lipid staining in those Kupffer cells, suggesting that those Kupffer cells was acquiring cholesterol and lipids through the use of those scavenger receptor, which recognize modified lipoproteins. To truly demonstrate that the observation that we were doing on those Kupffer cells were truly due to an increase in plasma cholesterol and not a secondary effect of changing, for example, the microbiota, you have heard from Karine talk, we used ezetimibe, which is a specific inhibitor of intestinal cholesterol absorption. So those LDL receptor knockout mice were fed either chow, a high cholesterol diet for 4 days, a high cholesterol diet for 4 days, plus this ezetimibe inhibitor. By doing so, blocking cholesterol absorption, you don't have any more elevation of plasma cholesterol, even though those animals are fed with the high cholesterol diet. And without the elevation of plasma cholesterol, Kupffer cell count remains steady. They do not proliferate, and they do not show any increase in lipid staining. So really showing that what we were absorbing was due to elevation of plasma cholesterol and not an indirect effect of dietary cholesterol-induced gut microbiota dysbiosis, for example. So we went out because those were short studies, only 4 days. So we wanted to know what's going on if hypercholesterolemia was maintained. So we fed the mice for a longer period of time, for 3 weeks. So if Kupffer cell count were increased, we just saw in the early days, then after 3 weeks, the Kupffer cell count now decreased. If you look at the composition of this Kupffer cell by flow cytometry, under chow condition and high cholesterol 4 days condition, this is pretty much the same profile. Those Kupffer cells express the TIMD4 receptor. And now when you look after 3 weeks of diet, there is now a second Kupffer cell population that it is called MoKC, and those MoKC are monocyte-derived Kupffer cells, and they're very characteristic with no expression of TIMD4. Okay, so if we looked at how those Kupffer cells behave during cholesterol diet, so embryo-derived Kupffer cells expand and then contract after a few days to diminish, while at the same time, those monocyte-derived Kupffer cells progressively engraft the liver. And at the end, after 3 weeks, we have about 50% of the Kupffer cell pool that is composed of monocyte-derived Kupffer cells. So what triggers EMKC pool contraction during this hypercholesterolemic condition? Is it EMKC suffering? So we look at the lipid loading, and we could see easily that this lipid loading is increasing between the first days to 20 days of diets. And this was clearly associated with increased oxidative stress with detecting mitochondrial ROS, reactive oxygen species, in those Kupffer cells. And it is pretty well known in the atherosclerosis field that accumulation of cholesterol in macrophage can trigger mitochondrial oxidative stress and favor death of macrophages. So we then wonder whether protecting those EM Kupffer cells to apoptosis could eventually have a beneficial effect. So this is what we did using one mouse model we have in the lab, which are transgenic mice overexpressing the mitochondrial anti-apoptotic protein BCL2. And this is specific, this overexpression is specific in the macrophage with a CD68 promoter. Those mice were crossed in a CCR2 knockout background, irradiated and transplanted with a bone marrow with bone marrow cells which are GFP positive, allowing us to then to do monocyte lineage tracing. Monocyte deriving from the bone marrow would be GFP positive. Those chimeric mice were then injected with an AAV9 encoding a PCSK9 protein to allow us to induce hypercholesterolemia in those chimeric mice by feeding them a high cholesterol diet. So we analyze the Kupffer cells of those animals with flow cytometry. And as you can easily see, EMKC were very few EMKC were positive for the GFP fluorescent protein, while MoKC were clearly a lot of MoKC were GFP positive, showing that they were derived from bone marrow blood monocytes. So then we measure the abundance of MoKC in those livers, comparing the livers of the mice that were expressing the BCL2 protein in their Kupffer cells. And as you can see, protected EMKC cells, mice with EMKC protected cells had fewer MoKC, and this was also true with GFP positive MoKC. So demonstrated that protection of EMKC against apoptosis with a specific targeting on the mitochondria reduces the generation of MoKC in these conditions. So what were the difference between EMKC and MoKC? First, we looked at the lipid loading, and this was done by flow cytometry, and you can see that body staining was lower in the MoKC as compared to the EMKC. This was also confirmed by sorting the two cell type and performing lipidomic analysis, and those MoKC had fewer free cholesterol and also less cholesterol esters. And this was associated, as one would have expected, with less oxidative stress in those MoKC. And another feature that those MoKC had higher capacity for proliferation as compared to the embryonic Kupffer cells. We also perform RNA sequencing of both cell type, and focusing again on scavenger receptor, and especially in CD36. 36 expression was lower in these MoKC at the mRNA levels, but it was also true looking at the protein membrane expression by flow cytometry. And then we evaluated whether the fact that those cells were expressing less scavenger receptor, and notably CD36, we injected LDL receptor knockout mice fed a high cholesterol diet with oxidized LDL, which had been labeled with a body P. Oxidized LDL are very specific modified LDLs which are recognized by scavenger receptor and notably CD36. And by doing so, we could see that MoKC were less able to uptake modified LDL, and then making sense of the fact that they were less lipid loaded as compared to the MKC. So to summarize this first part of the result, the EMKC pool homeostasis is clearly affected, perturbating during hypercholesterolemia induction. Those Kupffer cells acquire a lot of cholesterol. They become cholesterol loaded. This is partly mediated through the CD36 receptor that is able to uptake cholesterol-rich modified lipoproteins that are present under hypercholesterolemic condition. The Kupffer cell pool is expanded, and this expansion is mediated by proliferation that is triggered by the increased expression of CSF1 by stellate cells. And this occurs very rapidly in a matter of days. And then if the hypercholesterolemic condition are left maintained for a longer period, then this EMKC pool contract, and this is most likely due to oxidative stress going on into this EMKC pool, provoking their apoptosis, then liberating some room in the niche of those Kupffer cells so that monocytes can engraft and differentiate into monocyte-derived Kupffer cells. And those MoKC, monocyte-derived Kupffer cells, exhibit a lower lipid loading capacity that is associated with a lower oxidative stress present in those cells. And these MOCAC have also a higher propensity to proliferate, so providing them an advantage against EMKC to survive in those conditions. So after this observation, we wanted to evaluate what were the consequences of EMKC to MOCAC replacement. So to do so, we took advantage of a mouse model we had in the lab, which are the CD207 DTR transgenic mice that were crossed into a LDL receptor knockout. These DTR transgenic mice, they overexpress the diphtheria toxin receptor on Kupffer cells, okay? So if you inject them with diphtheria toxin, here DT in blue, the binding of DT to its receptor will trigger EMKC death by blocking translation. Control conditions were done by inactivating the diphtheria toxin by heating the toxin. Both groups were then placed on the high cholesterol diet for 8 days. Then we performed flow cytometry analysis of the Kupffer cell population, and then, as expected, EMKC pool was decreased due to the injection of the diphtheria toxin. And at the same time, the DT-injected animals exhibited a larger amount of MOCAC that were recruited to fulfill the niche after EMKC death. So at the end, after 8 days of this condition, MOCAC represented almost 60% of the KC pool, while the overall KC count were unchanged. Surprisingly, when we look at plasma cholesterol, we could observe that plasma cholesterol were higher in the DT-injected group. So we wanted to know whether this was only transitory or this effect could last for a longer period. So we performed a different experiment, and notably we left the animal fed on the high cholesterol condition for 4 weeks and repeated the experiment, analyzed the Kupffer cell population. Again, EMKC were decreased as expected in the DT-treated group, while MOCAC engrafted the liver. So you had an enrichment in MOCAC cells in the Kupffer cell pool. Kupffer cell counts were unchanged between the two conditions. And again, we could observe an increase plasma cholesterol level, suggesting that decreased EMKC and the replacement by MOCAC aggravates durably hypercholesterolemia. So to further understand what's going on, we performed transcriptomic analysis of those of the livers of these animals, and we performed a pathway enrichment analysis. And notably, we looked at transcription factors, and what we could see is that specific target gene for ERIXOR and ELIXOR transcription factor were decreased, and suggesting that there was a reduced ERIXOR and ELIXOR signaling in those DT-treated animals. And it is well known that ERIXOR and ELIXOR are key regulators of hepatic cholesterol and bile acid homeostasis. So to confirm this, we perform liver lipidomic analysis of the DT and BDT treated animals. And as you can see here, DT treated animals have had higher free cholesterol content and higher cholesterol ester content in the liver. And this was associated, as expected, with an increased output of fecal bile acid in those animals. So to finish, we wanted to know whether this effect of this replacement of EMKC to MOCAC had any peripheral consequences, and notably we looked at atherosclerosis. That is the connection between hypercholesterolemia and atherosclerosis is well known. So we performed already staining of aortic sinus of these animals to stain the lipid lesion in those mice. And in both females and males, we could see an increased lipid lesions in both in DT-treated animals. So to conclude on this study that was published less than a year ago, so there is some kind of a connection between hypercholesterolemia and Kupffer cells, where these Kupffer cells get lipid-loaded, cholesterol-loaded, and most likely through the uptake of modified lipoproteins, which leads to a lipotoxicity, oxidative stress in those embryonic Kupffer cells. And then there is a pull adjustment with the recruitment of monocyte-derived Kupffer cells. This pull adjustment will rewire liver cholesterol metabolism, potentially through LXR transcription factor activity. Then this leads to aggravate hypercholesterolemia and leads to increased atherosclerosis. So I thank you, and this is some of the people that participated to this study, and this is the grant agencies that help us for these studies. So thank you very much for your attention.
Okay, Thierry, thank you so much for your great talk. Quite interesting. The question that I was constructing in my mind was time by time answered by you. And it was very interesting to see that when you block the proliferation of Kupffer cells in the context of high cholesterol diet, it getting roast. You have more atherosclerosis, you have a higher level of cholesterol. And do you think it's, as I was discussing at the beginning, do you think that this is another example of how immune is important to modulate metabolism, in this case, lipoprotein metabolism?
Yeah, we have now more and more example of the connection between immunity and metabolism, especially under pathological condition. This study has been conducted under hypercholesteremic condition, which was a very specific focus. And this is why also we use those LDL receptor mice, which are very specific. They increase cholesterol and mainly cholesterol. And but you also have many papers that have been published, and one specifically by Emmanuel Gauthier, which lead our team.
Yes.
Under more condition targeting the liver, MASH condition, where there is not only an increase in cholesterol, but a triglyceride in those livers with a high lipotoxicity in this condition. And we could also demonstrate a link between those Kupffer cells and the impact of the pathology, where Kupffer cells, the replacement of those ENKC to MOKC had a burden into the inflammatory condition, and where the liver was less capable of storing lipids, which is the main role of this liver in this MASH condition. So yeah, there are many more examples now of how immunity and macrophages, which are really the cells we're interested in, and this connection is really what we are working on and interested in.
Yes, very good to see one more example of this interaction. And I have a question for you here from Marta Citelli, that is right now working in your team. And
okay.
she thank you for your talk first, no? And ask, do you think this may be related with ferroptosis?
This is not something we specifically looked at. This is I don't think it is really linked to ferroptosis in those specific condition and more apoptosis. The relationship between cholesterol accumulation in macrophages has been studied for years in the context of atherosclerosis, and it is known that accumulation of free cholesterol is really a trigger of ER stress and apoptosis of those macrophages. And this is not linked to ferroptosis, which is really a different DES pathway. And this DES pathway, we are studying it also under different condition, and in particular in condition of iron loading, which can occur in those cells since they are very eager to phagocyte dying erythrocytes. And you know, uptaking a large amount of erythrocyte every day will lead a large amount of iron in those cells. So I don't think this is ferroptosis in this specific condition, but really linked to cholesterol accumulation. Although we did not specifically, for example, try to inhibit ferroptosis and see whether it could eventually block the contraction we would see after a longer period of hypercholesterolemia. But this is a very good point. Maybe we should try that.
Yes, she is close to
Thank
you.
you,
You
Marta.
can discuss, you can discuss personally about it. So, let me check here. I have more question from Aline. Nice presentation. Though your finds suggest that hypercholesterolemia primarily disrupt Kupffer cells' self-renewal capacity, or does it actually recruit monocyte-derived macrophage through inflammatory signaling?
Alors, the signaling for monocyte recruitment is most likely due to the fact that the niche of those Kupffer cells become empty. Whether the signaling is due to an inflammatory signaling, I don't really know. It has been demonstrated under a very specific condition where you really deplete acutely Kupffer cells, that there is a specific signaling that is notably a CCL2 dependent associated with TNF alpha production. I am not sure this is the case here in this condition. We tried to repeat this experiment into CCR2 knockout animal, and we didn't see a default into a monocyte MoKC recruitment. Because this is not like what has been performed in the past, where you really acutely remove every Kupffer cells. This is a different condition. Here, you have a progressive diminution of Kupffer cells. So the niche become available for monocytes to enter and differentiate. And since those monocytes, those MoKC have advantages to stay as compared to EMKC, since they load less lipid, since they proliferate better, and if they die, then another monocyte can enter and differentiate. So this is the reason those MoKC are recruited.
Okay. Great, Thierry. Thank you so much. I have here one more question, but it's from Laura, and she's also close to you, and I will ask her. Let me. I know that we are late, but I will. It is the last one, okay? So she said it's a beautiful data. Thank you very much. Have you identified any signal that could induce the driving of MoKC and their further replacement over the EMKC? Could that be targeting to prevent the progression of disease in higher HC animals?
No, we have not been able to do so, to block MoKC recruitment. And we were not able, except for what I've told you, this was not CCL2 dependent, since we demonstrated through CCL2 knockout that was not the case. So yeah, that would be nice to do so.
Yes, of course. Okay, so thank you so much, Thierry.
Thank you for the
Great
invitation again.
presentation. I'm happy being with you here this time. Thank you so much. And then now we have a little break. And for the next talk, I will invite you to come back at 11:10 so we can drink water or whatever and come back at 11:10. Okay? Thank you so much. See See you in the next 10 minutes.
So good morning, afternoon, or night to everyone. Thanks for the kind introduction. It is a pleasure to be here in an international event developed by our research group. Today, I'm going to present a topic that connects immunology and metabolism, the role of eosinophils in metabolic homeostasis and obesity. To start, I expect to answer a provocative question. Could eosinophils be unexpected allies in our fight against obesity? We usually ignore them in metabolic studies, but today I hope to change your perspective. We are all well aware of the context. We are facing a global epidemic. The prevalence of obesity and overweight is growing worldwide. And this isn't just a static issue. It is a massive public health crisis. Obesity is leading risk factor for the chronic diseases that killed most people today, including type 2 diabetes, cardiovascular diseases, and several types of cancer. This creates a significant economic and social burden in our healthcare systems. Therefore, there is an urgent need for new strategies, mainly therapeutic strategies for that. We cannot think only about eat less, move more. We need to deep understand of the pathophysiological mechanisms related to obesity, specifically the inflammatory mechanisms, to develop effective treatments. So let's talk about the protagonist of our story, the eosinophil. Under normal conditions, these cells are actually quite infrequent. They represent only about 1 to 6% of total circulating leukocytes. If you took a blood test, you might only see 20 to 500 cells per microliter. Because they are so rare, they were often overlooked in metabolic tissues. They originate in the bone marrow from hematopoietic stem cells. They undergo a complex maturation process before being released into the bloodstream to perform their defense functions. Traditionally, eosinophils are mediators of allergic response and defense against parasites. They have been known like this for decades. However, looking at this diagram, we see that the modern view is wider. Eosinophils are not just soldiers in the immune systems, they are regulators. They interact with mast cells, T cells, and epithelial cells. They are involved in tissue remodeling, repair, and in a little bit more than 10 years ago, they were also described to be involved in metabolic homeostasis in adipose tissue. The paradigm shift really happened around 2011 with the paper from Wu and collaborators. Wu and collaborators published in Science. They discovered that eosinophils are resident cells in adipose tissue. This was the starting point. They demonstrated these cells in the adipose tissue, as you can see here in the histology. Also, they used DATA knockout mice with the ablation of eosinophils, as you can see here, and also transgenic IL-5 mice that super express the eosinophils to be demonstrated, as you can see in the letter D here, the presence or the exacerbation of eosinophils or no presence of these cells using the SIGLEC-F marker, here demonstrated in green. But more than just the presence, they also show that mice-fed high-fat diet, the eosinophils are reduced, and the number of them in the adipose tissue is inversely correlated with mouse weight. So the discovery that eosinophils are normal components of adipose tissue was the starting point for investigating their role in metabolism. In this work, they also showed that these cells are essential for maintaining glucose homeostasis. The superexpression of IL-5 transgenic mice reduced adipose tissue mass and improved glucose intolerance. But the lack of eosinophils in DATA knockout mice fed high-fat diet exacerbated adiposity and glucose intolerance, compared with wild-type mice fed the same diet. Then, when they looked at adipose tissue, they found that eosinophils were regulating the metabolic environment. This concept was further supported in the next years, showing how this immune balance orchestrates metabolic health. As you can see here in this figure of a review in the next year, 2012, they demonstrated an adipose tissue in lean state with the presence of eosinophils, Th2, and Treg cells, contributed to the production of anti-inflammatory cytokines that maintain the M2 macrophage phenotype and insulin sensitivity. However, with development of obesity, these cells are reduced and monocyte recruitment to adipose tissue increases, along with an increase in pro-inflammatory cells such as M1 macrophages. This leads to increased production of pro-inflammatory cytokines and insulin resistance. Studies continue in mice and mainly reinforced the idea that eosinophils participate in glucose metabolism and the expansion of M2 macrophages in adipose tissue. Also, the research advanced to show that IL-33 and innate lymphoid cells type 2 are the upstream regulators that sustain the eosinophils population. And among the last different functional associations of eosinophils in mice was that they are essential for beiging or darkening of adipose tissue through the production of IL-4. This fascinating function was discovered by Q and collaborators in 2014. And this process, where we have the white fat that's more related with energy storage, takes on characteristics of brown fat that is more related to energy burning. You can see this clearly in this histology in white-type mice, here in this part. Cold exposure induces browning, the darkening and staining. In the IL-4 and IL-13 knockout mice, and also the mice deficient for eosinophils, this process is lost. So they cannot generate heat efficiently. But the treatment with IL-4 in mice fed a high-fat diet increases beiging. And the mechanism is interesting. They put the idea that eosinophils release IL-4 stimulating M2 macrophages to produce catecholamines, and these catecholamines act directly on adipocytes to increase the CP1 expression and thermogenesis. Despite knowing that eosinophils are important, exactly how eosinophils control the expansion and remodeling of adipose tissue was an open question. This led our research group at UFMG to investigate the hypothesis that eosinophils regulated this expansion by modulating specific inflammatory pathways. And why that? Adeline already explained much more than I explained here, but previously our research group demonstrated that ablation of receptor of platelet activating factor reduced a lot of production of inflammatory mediators, and this leads to an exacerbation of adiposity. Then, in the beginning, we would like to investigate how eosinophils participate in the adipose tissue remodeling. And this work resulted in our publication in the journal Metabolism in 2023. Eosinophils protect from metabolic alterations triggered by obesity, together with our collaborators in Sorbonne University. And to test this, we use DATA knockout mice. And just to remember, these mice lack eosinophils. And then we compare them with white-type biopsy mice. And what is different from Wu and collaborators in 2011? As you can see here, they only demonstrated the effect of these mice upon a high-fat diet challenge. Then let's look at our results. First, we'd like to know what are the changes in adipose tissue due to the lack of eosinophils. And even on a control diet, look at here the adiposity graph, the eosinophil deficient mice, that is the black bars, had a significant more adiposity than controls. So the adipocytes in deficient mice are also much larger, and we observed a significant hypertrophy of these adipocytes associated with increased leptin levels, no alterations in serum free fatty acids, but with a reduction in basal lipolysis by adipocytes in culture. This suggests that without eosinophils, the fat tissue cannot remodel properly, leading to larger and maybe stressed fat cells. Now I'd like to know what are the metabolic changes due to the lack of eosinophils without a challenge with diet. And metabolically, the picture is quite clear in relation to eosinophils. The deficient mice have a much higher peak of glucose tolerance test, so they are glucose intolerant. They present an increase in glucose and insulin in the fast condition in relation to their levels, and consequently their HOMA-IR index, which measures the insulin resistance, is much higher. And associated to that, we observed a reduction in adiponectin serum levels and increase in resistin levels. This confirms that the absence of eosinophils leads to, I can say, a diabetic-like phenotype. And this was previously also demonstrated their huge participation in glucose metabolism. This is perhaps the most interesting finding regarding the inflammation. We analyzed the cytokine profile in this tissue. We'd like to know what are the inflammatory changes in this context of lack of eosinophils. And as expected, we saw increases in pro-inflammatory markers like TNF and IL-6. However, you can see here in IL-13, we saw a massive increase in this cytokine in the deficient mice. This seems paradoxical because IL-13 is usually good, but our interpretation is maybe that the tissue is trying to compensate. The body senses that the inflammation and produces IL-13 to try to trigger M2 polarization, but because the lack of eosinophils, this signal fails to resolve the inflammation. Could be a compensatory but an ineffective response. Also, these knockout mice demonstrated an increase in the role in leukocytes in the adipose tissue, and that appears to be related with the presence of neutrophils and pro-inflammatory macrophages, indicating that in this context, they are fighting against a pro-inflammatory environment. So to summarize the animal data, eosinophils are key protective pieces in the metabolic puzzle. They regulate adiposity, support insulin sensitivity via IL-4 and IL-13 axis, promote beiging, and prevent excessive inflammation. However, despite these robust findings in animal models, more research is needed to fully elucidate their role in humans and their therapeutic potential. Then take home message about this part. Eosinophils are much more important than previously thought, playing a key protective role against obesity in experimental models. The challenge now is understanding how to leverage this knowledge for human health. So I still didn't finish. Now we must ask, does this translate to humans? Here is the challenge. Studies like this one by Musa and collaborators found increased eosinophils presence only by histology, so it's quite simple. In the subcutaneous adipose tissue of patients with metabolic syndrome. This contradicts our mouse data, where low eosinophils cause the problem related to metabolic alterations. So in humans, they seem to be associated with the pro-inflammatory state. So to my knowledge, this was the first study in 2019 to try to associate eosinophils with adipose tissue in humans. But in 2023, in the paper that we published by our research group in Metabolism Journal, we look at the human mental tissue correlations of eosinophils markers like CCR3, IL-5RA, and also CGLAC8 with glucose metabolism. Tissues are obtained from patients with obesity that underwent bariatric surgery. These markers often correlated positively with each other, just as a validation, but they also correlated positively with Kupffer, IRS1, and adiponectin in the adipose tissue. So human data confirms the results obtained in mice regarding the glucose metabolism, showing this relationship between eosinophils and a good metabolism in relation to glucose. But a very recent study published by Hernandez and collaborators in 2024 increased this history of eosinophils in humans. They showed by flow cytometry the amount of eosinophils in subcutaneous and non-mental adipose tissue in lymph and patients with BMI more than 30. And as you can see here, with the patients with obesity, eosinophils content was significantly reduced when compared with the lean patients, independent of which adipose tissue it is, subcutaneous or mental. Also, they evaluated the expression of taxine, that it is a marker for eosinophils, and the same result was obtained. And also, individuals with obesity had less IL-4 expression levels in stromal vascular fraction in subcutaneous adipose tissue, and also in plasma. And they also demonstrated that eosinophils in subcutaneous adipose tissue is also inversely related to BMI and HOMA-IR. And they suggested a different mechanism involving leptin. In lean individuals, here in the left, a higher number of eosinophils would allow the IL-4 signaling, keeping leptin in a low level. So the insulin sensitivity and also the low inflammatory profile is preserved. However, in the case of obesity, there is a low amount of eosinophils, low IL-4 signaling, and this will increase the synthesis and secretion of leptin. And this hypercholesterolemia would help to establish a pro-inflammatory milieu, driving insulin resistance. So we could think, is there a future for eosinophils as a therapeutic target? Potentially. We could think about strategies to restore the function of the cells in obesity, use molecules that mimic their beneficial effects, like target IL-4, IL-13, the liver. But open questions remain. How can we specifically increase beneficial eosinophils in adipose tissue without causing side effects, for example, allergies? And what biomarkers are used to identify individuals who would benefit from these therapies? Also, while eosinophils are clearly protective in mouse models, the challenge now is translating this biology into safe treatments for humans. So the research about eosinophils opened doors for new therapeutic ideas, but there is still a way to go before this becomes a clinical reality. I'd like to thank my partners in the Immunometabolism group, the collaborations involved at ReFMG, our international collaborators, and our funding agencies. And thank you very much for your attention.
Great, Marina. Thank you so much. So nice to hear you telling you this history. And let me check here. We have a question. I have
or
a,
not? No, I closed. No, stop share. Sorry. So now.
okay.
Yes.
Okay. Do you think that this effect of eosinophils to control fat mass expansion may be related with their function as a pro-fibrotic cells?
Oh. I never thought in the sense of to contain the expansion. I always thought about the other side, when they are maybe increased when they lose weight, so they could, for example, contribute with fibrosis. But maybe it's a good question that depending of the amount that you would have of eosinophils, they would control to not have like a huge amount of adipose tissue, because they got, yeah, nice question. I never thought in this sense, in the other side.
Yes, but it's
It
one
will
thing
not
we
be
should.
so difficult to try to see this, because it would depend only just to do maybe a correlation with fibrosis in the size of the mass of adipose tissue and this sense. So I try to see if they relate to that.
Yes, we should think about it and check. Even in for human data, we can talk with Genevieve after.
Oh yeah,
and
we
check
have already some idea in relation to already the presence of the in obesity patients, but I didn't think in the sense to reduce the mass of adipose tissue depending of the type of in the amount of cells that you have. So in the I complement the
Yes.
idea, so it's nice.
Okay, Marina, we have a question here for you from Ana Flávia Martins. Very interesting. Considering this, would there be an ideal percentage for serum eosinophils?
I didn't understand there. Would you?
She is asking whether do you think that the number of circulating eosinophils can predict?
Ah, okay. Wow. At this that we were discussing, for example, I believe that is not so explored. In fact, I added in my presentation most of these studies. I don't know if it lack one or another, because they are not so explored in the sense of adipose tissue. But one thing that could be thought to be done is, for example, to try to see if people that are not so, they have like a difficult to gain weight or something like that, if they have an increased amount of eosinophils, much more than others. I know that they are presented in a low amount in the serum, but at least we could try to range them in different categories and see, for example, these people with, I don't know, with a severe obesity, they have less eosinophils, but others in the middle have a little bit more, and try to stratificate in eutrophic people, and mainly people that don't have difficulty to increase weight. Why they have these difficulties? Because they have much more eosinophils. This could be something to try to determine which percentage in relation to body weight would be interesting. But at the same time, eosinophils participate also, as I put in the slides, in allergy, and also allergy processes, and also in fibrosis. For example, in lung diseases, they increase a lot fibrosis. So it would be, for example, if we try to manipulate the amount of them, the risk would be just to have side effects effects in relation to the other functions that they have. So, but for example, could try to see people that generally have allergy, low levels of allergy, if they are lean or not, or this kind of stuff.
Yes, it deserves to be explored,
Yeah.
no? And I will change to Aline question because it's related of what we are discussing here. She said, amazing, Professora Marina, not sure if this makes sense, but there is a safest way to increase eosinophils in fat tissue without increasing allergy risk.
This is still something that would be interesting to test. For example, in literature, thinking about mice, there are some possibilities, for example, trying to increase, as I showed you, in transgenic mice with IL-5, but there are other ways, for example, injecting IL-5, and this would be also not just only the transgenic as like a therapeutic way. It could be a guess trying to increase and not to be too much, but at least, for example, if you think about the obese people or people with obesity, that they have a low amount of eosinophils, maybe for them, this increase would be interesting. As transgenic mice for IL-5, they have a reduction in the adipose tissue.
Yes, okay. And one question from Joyce. Are there alteration in eosinophils activity and abundance during weight loss?
Is also not so explored, but there is a paper that did not evaluate exactly obesity, but they were evaluating senescence and trying to see the intermittent fasting effect. And they observed with intermittent fasting, they did like a 2 days feeding and 24 hours fasting, and they observed an increase in the presence of eosinophils. So in the sense that if you lose, because they demonstrated a loss of weight and the adiposity, but not in the sense of in the context of obesity, but they could see an increase in these cells, and they related that it could be interesting strategy to increase eosinophils. So I believe that in this idea of weight loss, just one thing that we need to take care is about also the other functions of eosinophils that could, for example, exacerbate in the condition of stress, I don't know, and we have the fibrosis in the adipose tissue, and this could maybe lead to an increase in the presence. The re-increase in the presence of these cells could exacerbate their function and contribute to fibrosis, but just a guess, we don't know, still don't know.
Oh, yes. Okay, Marina, we have to go further to our program. Thank you so much for your amazing talk. And thank you. And now I will invite Marta. Are you ready, Marta?
Yes.
Yes. You can start to share your screen. Thank you so much for coming. And Marta is here to conclude this section. And then we are honored to have you here. Marta is a colleague, is a partner in our research group, and she is a professor at the State of University of Rio de Janeiro. She has a solid academic trajectory with a research focus on iron metabolism. That it's a very interesting field to be explored. So thank you so much, Marta. Nice to see you and Amanda, because I can see Amanda. So nice to see you both. And that's it. Okay, thank you.
I'd like to. Are you seeing the presentation? Is it
Yes, I'm seeing, but I'm seeing something weird here because I can see the next slide.
Yes,
yeah.
I can.
We're gonna change.
Okay, but
Nova
okay,
Bangladesh.
that's okay. That's
Okay,
fine now.
works better. Thank you.
Okay, okay. No.
Sorry.
I think, yeah, okay. So first of all, I'd like to thank you for the opportunity to be here in France through this CAPES-COFECUB project. So Adaliane, thank you very much for this opportunity, and thank you Geneviève and Professor Karine Clément. So I'm going to talk about iron metabolism, and to start this, I'd like to explain the importance of iron in life. So the origin of life may have begun with iron because our Earth was plain of iron dispersed in the oceans. So in the Archean period of Earth history, iron was dispersed, and iron, because of its possibility of changing electrons, it can activate the oxidation of many molecules, and it already has a very important role in like a catabolism, like a catalyst mechanism of beginning the life. So life, as we know in Earth, may have begun with iron oxidation and reducing property. So iron functions as a perfect electron carrier. So it can charge and discharge, what's very important for many metabolism and many catalyst actions. And iron is involved in many roles in mammalian organisms. The main roles are listed here, and the oxygen transport is a very important role. Electron transport and energy metabolism also, because ATP synthesis is dependent on iron, and DNA replication and repair. But iron participates in many other functions, but here we are just talking about these three to understand the importance of iron in life. So iron participates, is included in the hemoglobin, that is a protein that is carried by red blood cells, and is a protein that carries oxygen throughout the organism. And the other function, very important, is the ATP synthesis that is done by mitochondria, because all these proteins that are present in the electron transport carrier, electron transport in the mitochondria, all these proteins are iron proteins. So iron is very necessary for ATP synthesis and also for DNA replication and repair. So seeing this, I think it's clear the importance of iron in life. But although it is very important in life, it's also iron is also toxic, because iron participates in the Fenton reaction, producing hydroxyl radical and other radicals and other molecules that are reactive. So although iron is very necessary for life, it's also very toxic. Because of this, and because of because mammalian organisms do not detox iron, do not excrete iron in a manner that's regulated. Because of this, iron must be absorbed in a very carefully system that is regulated by hormone. So there is a hormone called epsidin that blocks iron absorption. So epsidin hepcidin has, as some people say, hepcidin ligates on ferroportin. That's a protein that's present in enterocytes and also present in macrophages, also in Kupffer cells. What hepcidin does is to bind ferroportin and to blocks iron exportation to the plasma. Here in enterocytes or in macrophages, the mechanism of action of hepcidin is the same, is to ligate ferroportin and to blocks iron exportation. So this is so iron homeostasis is very fine regulated, and it's regulated in a manner to that in a way that iron can compose the iron that is lost. So in the absorptive mechanism that we have, we control the absorption to have about 1 or 2 milligrams of iron per day to compose this loss that we have every day. Most of iron we use comes from the iron recycling macrophages, and this regulation is very important. So because of the characteristic that iron has, that it can oxidize many molecules. So because of this, iron challenges redox system, and cysteine is an amino acid and also methionine that composes glutathione, and glutathione is a substrate for glutathione peroxidase 4. That's one of the key enzymes necessary for lipid detoxification and to don't permit that iron can be toxic. So glutathione peroxidase 4 is very related to iron homeostasis. And this enzyme, glutathione peroxidase 4, is a selenium enzyme. And as I have said before, glutathione is a substrate for glutathione peroxidase 4 and is used here. So when there is less cysteine or methionine or even selenium, because selenium is part of GPX4, when there is insufficient of these cells usually goes to oxidative stress. So iron poses this oxidative stress, and if we don't have sufficient antioxidant mechanisms, cells may go to ferroptosis. So ferroptosis is a kind of cell death that is initiated by free iron, and also by lipid peroxidation. So lipid peroxidation that is induced by iron and that does not have a sufficient antioxidant mechanisms, these cells goes to ferroptosis, this kind of cell death. And selenium as part of GPX4 is a protagonist of this mechanism. So this redox homeostasis is very necessary to maintain health, but also it has been seen that ferroptosis in some way seems to be a native way of cell death that may regulate the cell death of those cells that must dead. So in cancer, for example, we hope that cells that's going to a cancer phenotype, they usually can dead through this mechanism of ferroptosis. And in some kinds of cells, the control of oxidative stress is very important. So here we observed in red blood cells of sickle cell patients that selenium has a very important role in maintaining their health. And when selenium is diminished, cells emolysate more easily. So it's important to maintain this antioxidant equilibrium, and we have many plant bioactive compounds that can deal with this challenge. So here so in our group we are studying substances that are present in foods that can control iron homeostasis. And we have observed that, for example, in a study that we did in our lab, that broccoli sprouts that are food which contain high levels of sulforaphane. Sulforaphane is a substance that activates NRF2, that is a transcription factor that activates antioxidant defense. So we observed that broccoli sprouts that are rich in sulforaphane modulates iron homeostasis. And here, when we used high levels of sulforaphane, we had an increase in hepcidin production. And we also observed that in this condition of 50 micromolar of sulforaphane, we had diminish of ferroportin. So this may have induced iron accumulation in these cells that received 50 micromolar of sulforaphane, and it may have produced, this excess of iron may have produced ROS production, an increase in ROS production, and the cell viability was diminished in this case. So what we are seeing here is that when we treated cells with sulforaphane, we had an increase in hepcidin that may have blocked ferroportin exportation of iron, and this accumulation of iron may have initiated cell death. So here in Sorbonne, with this group, what I'm interested in investigating is the role of iron in adipose tissue remodeling. So I'd like to thank these researchers that participate in this study more directly. And what we are starting doing is to investigate the relationship of iron metabolism in the adipose tissue. So as the group works with fibrosis, and as fibrosis is a mechanism that is being revealed and being studied in the adipose tissue, and as it involves ROS production and antioxidant mechanisms are participating in this process, and also oxidant mechanisms. So what we want to see is if iron accumulation can drive fibrosis in adipose tissue. This study here demonstrated that iron accumulation drives fibrosis in other tissues like liver, kidney, and also in heart. But what we want in lungs, but they didn't study iron accumulation and fibrosis in adipose tissue. So our hypothesis is that iron homeostasis is a key determinant of adipose tissue fibrosis, potentially through ferroptosis, and contributes to healthy adipose tissue remodeling during periods of fat mass gain and loss. And the other hypothesis is that iron availability regulates mitochondrial biogenesis, as iron participates and is part of all that proteins of electron transport in mitochondria. So it may regulate mitochondrial biogenesis, thermogenesis, and basal energy expenditure in human adipose tissue. So I'm sorry, there is this configuration here, but it's okay. So in this study that we are beginning to do here with the serum of patients that had undergone to bariatric surgery, we have now this design of a study evaluating them in the moment, in the basal moment, and after weight loss, 12 months after surgery. And what we have, what the material that we have and we can explore to test this hypothesis is serum, adipose tissue, and the data bank that had already been collected. And also we have some slides to do per-Prussian staining. That's a kind of stain to detect iron. So we are doing this at the moment 0 and at 12 months later. And we want to test this hypothesis. We cannot, we are going to do some associations because the intervention was weight loss. So the direction of this question here is we are just going to do this association, but we have some preliminary results that we observed in the adipose tissue that undergoes fibrosis when we stained this tissue with per-Prussian. We observed some signals that are blue. We can see here it's very dark because I think there is iron accumulation here, but it's somewhat blue and it indicates iron in the tissue. And we are thinking that it may be related because this pink color here indicates fibrosis, and in the same tissue we observe iron accumulation. So it may be, it's a very preliminary result. It's just to start. And we also did some associations, and we observed that a direct relationship, a direct association between the rest energy expenditure calculated by indirect calorimetry or by DEXA, and we observed a direct association with rest energy expenditure and some indicators of iron metabolism. So in our lab, there are 2 students in Brazil participating in this study, and the design of their thesis is like this, in which we are submitting the animals in a high-fat diet for 2 months, and these animals are receiving high-fat diet with deferoxamine, that is a iron chelator, or a supplement of iron, or even the sprout, the broccoli sprout extract. I'm sorry, because this broccoli sprout extract is rich in sulforaphane. So they are gaining fat mass like this in this design, and they are going to lose weight. We we have not reached this phase of the study, but they are they are receiving food to have this moment, and after that they are going to regain fat mass, and we intend to observe the changes that are being initiated in these animals when we offer more or less iron, and when we offer some substance that we know that regulates hepcidin and regulates iron metabolism. So this is what we are doing now. And I'd like to thank people that are involved in this project. And here in France, I have already thank you. And in Brazil, these are the students of our group. Carolina Soares is doing her thesis on it, and also Mateus. And this is Rio de Janeiro. Thank you very much.
Thank you, Marta. Rio de Janeiro is still beautiful, amazing, wonderful place. Thank you for your talk. And what I have to say is that I'm quite excited to see your data and the outcomes you can get, because to understand whether the iron is or not related with fat remodeling, it would be a very new field to investigate and a very exciting one. I have a question for you. Here is Marina. She's asking, the question is, more than a broccoli component, have you tested and observed good effects on iron metabolism with other type of food? Anything about obesity, if increased iron metabolism is related with fibrosis? Is there a paradox?
Oh, so we have already tested other foods. We have tested green tea, and we observed that green tea, when we used high levels of green tea extract, or even we use high levels of catechins that are present in tea, but not only the tea extract, but when we used higher concentrations, we observed the cell death. And when we used concentrations that are not so high, lower concentrations, we observed a phenomenon that was not cell death, but the opposite of this. We observed some proliferation in cells, and we related it with iron metabolism. So we observed that these concentrations provoked, these concentrations that dead the cells provokes accumulation of iron inside the cell, and it provokes ROS increase. So we think it may be related, and it may be a mechanism of cell death that perhaps is ferroptosis. We have some markers, and it seems to be related. In obesity, we have investigated it in human beings, and we observed that children that have high adiposity, they also have high hepcidin. It's not only our observation; other studies had already demonstrated it, but we saw that it modulates the obesity. That it's another way to see the relationship is that obesity modulates iron metabolism. Here we are trying to see if iron metabolism can modulate obesity.
Yes.
We can see in both ways. I don't know if answered everything.
Yes, yes, we have here. I have one more question for you, but I have others I can send for you, and you can reply for who are asking for it. But here, Thierry, he is asking here, does Marta know what is the effect of iron supplementation or deprivation on microbiota? Mouse studies, human data, and thus potential consequences on metabolic issues. I picked this
Yeah.
question because it's quite interesting.
Yes, it's
Thank you,
very
Thierry.
Thank you, Thierry. Yes, it's very interesting. I have seen some studies that demonstrate that iron supplementation it depends on the dose that is given, but it modulates microbiota, and it in children it may produce some undesirable effects in even in growth or in cognitive development. So iron supplementation is not something that does not provoke any effect because it modulates microbiota. And in obesity, we didn't see any study. I didn't see any study. I have investigated it with Carolina Soares, this student, because we were going to study this, but we had to change our plans. But we had investigated somewhat in this in obesity, and there are very, very poor information about this relationship. I think it's interesting.
Yes, yes, it's interesting. Okay,
Thank you.
thank you, Thierry. I will move. I have to, because we still have one short talk and to finish this section. And thank you so much. It's nice having you here, Marta. Thank you so much. And I will now move for the short talk. It's important having short talk because we can see the younger students sharing with us what they are doing. So it's quite exciting seeing this emerging research, what they are doing and what they can mirror the next generation of scientists. So for this, I'm warmly invite Joyce Herédia. She is a member of our Immunometabolism research group. She holds a master's degree in cell biology, and she is currently a PhD candidate in nutrition at the Federal University of Minas Gerais. Okay, Joyce, thank you.
Thank you, Dana. I will share my screen. 1 second. Can you see my screen?
Yes, but it is not in a full, yes, now it's.
Yes, yeah, perfect. Thank you. 1 minute. So, hello everyone. My name is Joyce. I am PhD student, and today I want to talk about bone metabolism and the association with the obesity. So, I would like to start with 2 facts. Technically, everyone is going to age. And the second fact is everyone is going to lose bone tissue. And this is will start especially during the aging. And because of that, it's so important to study the bone tissue and its structure. So the bone, it's, let me get a lazy on it. The bone, it's a calcified structure and can be divided in 2 parts, the sponge bone inside and the compact bone outside. And this structure can be changed through different cells in this environment. So the osteoclasts are the cells responsible for the bone resorption, the osteoblasts are the cells responsible for the bone formation, and the osteocytes are the cells responsible for coordinate the activity of the osteoblasts and the osteoclasts. So the cells is responsible for regulate of the bone metabolism through the secretion of a lot of cytokines. So the dysregulation of osteoblasts and osteoclast activity can cause impaired bone remodeling, and this a lot of times can be caused for a dysregulation of the activity of the osteocytes. And this impaired bone remodeling can cause a weight loss or increased risk of fracture. And there are a lot of causes of this impaired bone remodeling, such as lifestyle, medications, endocrine, inflammatory, and metabolic alterations, aging, and also obesity. So a lot of studies are trying to understand this relationship between obesity and bone alterations. And our group showed that the high carb diet can decrease the bone mineral density and also the bone volume. And it's important to know that this high carb diet can improve the fat, but not the weight of this mice. And this happens in the long bones, like the femur, but the same, we can see the same in short bones, like the jaw, and in this case, the same results. Decrease in the bone mineral density and the bone volume with the high carb diet. But on the other hand, other types of diet can have a different results in the bone metabolism. So the high fat diet cannot alter the bone mineral density and the bone mineral density associated with the bone volume. There are a difference between this both diet because the high fat diet can improve both the weight and also the fat mass, the adipose tissue. And in addition, in my work, I tried to understand the chronology of the bone alteration with this high-fat diet, this obesogenic diet, and we could see that have a decrease in the bone mineral density and also in the bone volume with the age of these mice, but not with the high-fat diet, with this obesogenic diet. But when we evaluated these cells in the bone environment, we could see the same for the osteoblasts. But it's interesting to know because the number of the osteocytes are we could see a decreased number of the osteocytes with this high-fat diet. So this fact made me curious about these osteocytes, and I want to know a little bit more about the morphology of the cells. So with the transmission electron microscopy, we could see changes in nuclear morphology of the cells during high-fat diet in obese mice, and also cytoplasmic lysis and increased osteocyte area. And don't have papers about this fact, these alterations in the morphology of the osteocytes during the obesity, but these alterations are related with senescence alterations. So we went to evaluate alterations in the bone environment about the senescence, and we could see an increased expression of a lot of markers of senescence in this environment, in the tibia and also in the bone marrow, and this for the specific marks of the senescence and also for the inflammatory cytokines in the all data with 16 weeks of high-fat diet. So in summary, we could see that the obesity can improve this weight, for example, with this high-fat diet, and this can have a protective effect against bone mineral density reduction. But the improve of the adiposity that this occurs, for example, with high-carb diet, can improve this bone loss. But the both situations, we have alterations in the bone quality in mice model. But in about human studies, so a lot of studies show a lot of studies with humans. It's more constant about the relation between bone and obesity. And for example, this systematic review and meta-analysis showed that increased bone mineral density in patients with obesity. And other studies show a positive correlation between the bone mineral density and body mass index, and a negative correlation between the total body fat and this bone mineral density. Despite these alterations in the obesity and the bone mineral density, are related in the literature that the obese people with obesity can have increased risk of fracture. This occurs because this type of patient have increased risk of falls, and this is associated with alterations in the bone environment, like increased adipocytes, cytokines, and reduced bone strength, and this can improve this risk of fracture. So this bone paradox during the obesity can be explained in two ways. The first one is the mechanical effect of the obesity. So an increase of weight can increase the bone density because the weight can affect the osteocyte activity, and these osteocytes can improve the activity of the osteoblasts and the number of the osteoblasts. And the cells are responsible for forming the bone tissue. But on the other hand, the metabolism and the inflammation associated with the obesity can decrease the bone density. This occurs because increased fat can improve this inflammation, and the inflammation are associated with osteoclast activation. In this osteoclast, it's responsible for the bone resorption. So an imbalance in this process is reflected in how this bone will respond. So for the take-home message, we could see that the bone outcome will depend on several factors. Obesity can increase or decrease bone mineral density depending on the stimulus. And in any situation, bone quality decreases and the risk of fractures increases. So I would like to thank for my research group and also for our sponsors. Thank you.
Okay, Joyce, nice to hear you. Thank you so much for being here, sharing your work with us. We are now, it's 12:20 in Brazil and 4 hours plus in France. And we have to stop for lunchtime. And so I will finish this section. Thank you again, all the speakers, Karine, Clément, Thierry, Marina, Marta, Joyce, for your kindly talk, for sharing with us what you are doing, have been doing. Thank you so much. And I will invite you to have lunch and come back at 12 p.m. in Brazil, for 12 for let's say to 6 p.m. in France. I know it's late at night, but I hope you can continue being with us for the second section of our meeting. Thank you so much. Okay, see you.
Bon appétit.
Merci. Bye-bye. Hello. Before we officially start, I would like to thank Simon and Amanda. I see you here. Thank you so much. Good to see you here.
Hello, Adaliane.
Hello, Simon. Hello. Nice to see you. Thank you so much. We have a lot of people exciting to see your talk about creatine system. Thank you.
Yeah, I have a technical issue. Just go on, please.
I have, okay.
I think it's fine now. Sorry.
Yes, the sound is
Okay,
much better now.
It's okay.
It's okay.
Yes, it's okay, Amandinha. Yes,
Sorry.
no problem.
Hi, everyone. Thank you so much, Adalien, for your invitation. So my name is, I can start?
I'm sorry, Simon. Who will host this section? It would be Marina.
Okay.
Marina Chaves. She's a professor here with us, and I shared with her. I stayed during the morning, and she will stay in the afternoon. I'm just I got in just to say hello to you and thank you because I realized that you were here preparing. But now it's the time at 2 p.m. here, for you 6 p.m. and Marina will be the chair, okay? Thank you, Simon. Thank you, Amandinha. See you.
Thank you so much.
Okay, hello everyone. Let's restart. In this afternoon, I'll be the chair. And before we begin, just a quick note, we encourage your participation. So please feel free to type your question in the chat at any time during the presentations, and I'll read them out for our speakers after each talk. So to start our afternoon session, it's my pleasure to welcome Dr. Simon Lecoutre. Simon is a member of the Nutriomics Research Group and a researcher at INSERM. After earning his PhD in physiology from the Université de Lille in 2017, he expanded his expertise internationally. He finished his postdoctoral fellowship at the Karolinska Institute and later at the UMRS in Paris, where he worked on adipose tissue remodeling and redox homeostasis. Simon, thank you for being here today, and the screen is yours.
Hello. Hi, everyone. Thank you so much for the introduction. So my name is Simon. So I'm a researcher in the lab of Karine Clément, and my work focuses on a sample ID, and I'm very sorry for that, how metabolism shapes the identity and function of adipocytes. And today I'm going to talk about the role of creatine metabolism in white adipocytes. And this project is a collaboration with my beautiful colleague and friend Salwan Magdazi from the Karolinska Institute. So, as you know, in the context of obesity, chronic energy excess push adipose tissue to expand, and this expansion can occur through two distinct mechanisms. You have adipocyte hypertrophy, in which existing adipocytes increase in size as they accumulate triglyceride in the lipid droplet, or you have adipocyte hyperplasia, in which new adipocytes are generated from precursor cells. When adipocyte size, here you can see in the y-axis fat cell volume, is plotted against body fat mass in the x-axis, one observes individual variability in mean adipocyte size for a same fat mass. This variability, as you can see here, reflects fundamental differences in the mode of adipose tissue expansion. Importantly, individuals whose fat mass increases predominantly through adipocyte hypertrophy, exhibit a higher risk of developing cardiometabolic complications. Indeed, hypertrophic adipocytes progressively lose their normal functional properties. As adipocytes enlarge, their phenotype shifts toward a state of partial dedifferentiation characterized by a reduction in the expression of key adipocyte-specific genes and protein that normally define and maintain adipocyte identity, such as perilipine, LPL, adiponectin, for instance. As a result, their phenotype gradually shifts toward a more fibroblastic-like state and become associated with the activation of a fibro-inflammatory program. In this altered state, hypertrophic adipocytes secrete elevated level of pro-inflammatory cytokines, such as TNF-alpha or IL-1 beta, and pro-fibrotic mediators. These signals are well known to impair both insulin and catecholamine responsiveness, substantially reducing the tissue capacity for safe and efficient lipid storage. Consequently, hypertrophic adipocytes develop increased basal lipolysis and heightened inflammatory secretum, which together promote the recruitment and accumulation of immune cells, such as macrophages, within the adipose tissue microenvironment. In parallel, excessive extracellular matrix deposition progressively stiffen the adipose environment, leading to fibrosis that physically limits the tissue ability to expand and limit adipocyte hypertrophy. Thus, once adipocytes reach their maximal storage capacity and can no longer safely accommodate additional lipid influx, lipids begin to escape the adipose tissue and accumulate in the peripheral organs, such as liver and muscle, for instance. And this lipid spillover drive ectopic fat deposition, lipotoxicity, and systemic insulin resistance, ultimately contributing to a persistent state of chronic low-grade inflammation. This sequence raises a critical mechanistic question. What drives hypertrophic adipocytes to adopt these fibro-inflammatory states? A major clue lies in metabolic stress. Excess nutrient exposure induce mitochondrial overload, mitochondrial hyperactivity, reflected by increased oxygen consumption and elevated production of reactive oxygen species. And this change generates local hypoxia in the tissue and in adipocyte and trigger metabolic reprogramming that alter adipocyte identity and promote fibro-inflammatory activation in these cells. However, the fundamental nature and initiating trigger of this metabolic stress remain incompletely defined. This leads to the central question guiding our research. How do adipocytes lose their identity and acquire a fibro-inflammatory phenotype during obesity? So one of our important ideas that I've developed with my colleague Salman Magdazi when I arrived in the Karolinska Institute in the lab of Michael Reeden. The idea is that metabolites themselves are powerful regulators. They are not just fuel, they act as cofactor or substrate for epigenetic enzyme. As you know, for instance, acetylcoenzyme A is a substrate for protein acetylation and histone acetylation that can open the chromatin and increase gene expression, for instance. And alpha-ketoglutarate, for instance, it's a cofactor for histone demethylation or DNA demethylation. So we therefore hypothesize that metabolic rewiring in obesity drives adipocyte dysfunction. So to test this, we profile, when I arrived in postdoc in Karolinska Institute, the metabolome of subcutaneous adipose tissue from 13 obese and 13 non-obese women. Among all detected metabolites, phosphocreatine stood out as the most strongly increased in obese adipose tissue. This suggested that creatine metabolism might play a previously unrecognized role in adipocyte dysfunction. What is phosphocreatine? So phosphocreatine is a high-energy molecule that allows instant ATP regeneration when cells face a sudden increase in energy demand. about half of our creatine comes from the diet, and the other half is synthesized by our own metabolism from arginine and glycine via two specific enzymes, GATM and GAMT. And it is very important to know that GATM is not expressed by mature adipocyte. So inside mitochondria, CKMT, creatine kinase in mitochondria, fleshly produce ATP from mitochondria, from the respiratory chain and ATP synthetase enzyme, to charge creatine into phosphocreatine, which then travel to the cytosol. There, so in the cytosol, enzyme like CKB, creatine kinase brain isoform, instantly convert phosphocreatine back into ATP and creatine, exactly where and when the cell needs it. This system, creatine metabolism, has been a really nice map in bone adipose tissue thanks to pioneering work from the group of Bruce Spiegelman and Laurence Kazak. So classic bone adipocyte is known for UCP1-driven thermogenesis. So UCP1 allows proton to leak across the mitochondrial inner membrane, uncoupling respiration from ATP synthesis. This creates a futile proton cycle, dissipating energy as heat. But broad adipocyte also relies on powerful UCP1 independent thermogenic pathways, and one of the most important is a futile creatine cycle. So in this pathway, CKB phosphorylate creatine, while TNAP, tissue non-specific alkaline phosphatase, hydrolyze phosphocreatine by releasing the phosphatine organic and creatine, establishing a continuous back and forth reaction that burns ATP without producing work. This constant ATP turnover generates heat and represents a major alternative thermogenic mechanisms independent of UCP1. However, the physiological relevance of creatine metabolisms in white adipose tissue had never been explored. To determine whether the phosphocreatine accumulation seen in obesity in the subcutaneous white adipose tissue reflect change at the gene level, at the gene expression level, we analyze transcriptomic data from subcutaneous white adipose tissue of women with and without obesity. And this transcriptomic data shows that CKB, the cytosolic creatine kinase responsible for converting phosphocreatine into ATP, as you can see here, was markedly reduced, a finding confirmed by immunofluorescence, where the CKB signal is strongly reduced in the subcutaneous adipose tissue from obese patients. Thus, a picture emerged: phosphocreatine rise while CKB expression and activity fall in obese adipose tissue. This led us to hypothesize that CKB loss lead to phosphocreatine accumulation. To test this, we silenced CKB in differentiated human adipocyte by scRNA. So we did the scRNA after 8 days after the induction of adipogenesis. CKB knockdown reduced CKB mRNA and protein levels and increased phosphocreatine without altering creatine abundance. We then validated this finding in vivo using adipocyte-specific CKB knockout mice, where CKB flux mice was bred with adiponectin CRE mice to deplete specifically CKB in the adipocytes, as you can see here with the qPCR. And as you can see also by the immunofluorescence markage, with a reduction of CKB expression in the CKB knockout mice. And this was associated by higher phosphocreatine level with the creatine level was unchanged. So this mirror exactly what we observed in vitro human adipocyte and human white adipose tissue. Next, we examine which biological pathways are altered when CKB expression decreases. To address this, we integrated two complementary datasets. Firstly, transcriptomic profiling of in vitro human adipocyte in which CKB was silenced. Cells were transfected with ACCKB at day 8 of differentiation, and mRNA sequencing was performed at day 11. Secondly, we combined this dataset with gene expression correlation from subcutaneous white adipose tissue of the 56 women, identifying genes whose expression level are associated with CKB mRNA expression in vivo. The convergence between these datasets was remarkably consistent. Loss of CKB downregulation strongly and reproducibly activated pro-inflammatory pathways, as you can see here. Inflammatory pathway and TNF alpha signaling pathway. Both in culture adipocyte and in human adipose tissue. Among the genes driving this inflammatory signature, CCL2 emerged as the most robustly induced in response to CKB silencing. Importantly, this molecular signature has clinical relevance. Women with low adipose CKB expression in the left of the graph exhibit higher CCL2 secretion by their adipose tissue. It's an ex vivo experiment. And this, we find a very negative and very beautiful correlation, and we find this correlation after the adjustment of the BMI, indicating that reduced CKB expression is independently associated with heightened systemic inflammation. To come back on the in vivo model in the mice, in the adipose site-specific CKB knockout mice, we observe that the loss of CKB directly promotes white adipose tissue inflammation. And here we focus on epididymal white adipose tissue on the visceral depots. This animal here in green show a strong increase in CCL2 expression and higher level of macrophages markers such as ADGR1, F480, and CD68 mRNA expression. And we can see here that in the knockout mice, there is a strong increase of chronic structure in the epididymal white adipose tissue. And here you have the quantification of F480 cells per feed that is strongly increased in the knockout mice. So mechanistically, we demonstrated that obesity reduced CKB expression in adipocyte, causing phosphocreatine to accumulate. Because the creatine shuttle become inefficient in the cells in obesity, mitochondria try to compensate by overproducing ATP, a response tightly coupled to increase glycolytic flow. But this excess of ATP has a crucial consequence. The excess of ATP will inhibit AMPK, an enzyme that is it's very well known for its anti-inflammatory function. Then, as a result, CKB depletion induces ATP overproduction that inhibit AMPK, and this will lead to adipocyte and adipose tissue inflammation, which in turn contribute to insulin resistance. This naturally led us to a key question. Why does CKB expression fail in obesity? So we have done a transcriptional screen that has allowed us to provide maybe partial answer, but an answer of this question. So we discover that ER stress, acting through the ER1 XBP1 pathway, recruits the DNA methyltransferase that we call DNT3A directly to the CKB promoter. Then DNMT3A methylate the promoter of CKB to silencing its expression. And what triggers this ear stress in adipocyte? Precisely the state of the hypertrophic adipocytes. Once enlarged beyond its physiological limits, it can no longer store fatty acid efficiently. It begins leaking lipids through elevated basal lipolysis and fail to re-esterify them properly. And this metabolic overload activates ear stress, which in turn shut down CKB via increased CKB promoter methylation. And this reduction of CKB expression will lead to adipocyte inflammation, and this will in turn promote the infiltration of immune cells in the adipose tissue. In the second time, we find another interesting thing that we observe precisely that in subcutaneous white adipose tissue in humans, CKB expression shows strong negative correlation with adipocyte size. And this correlation is independent of the BMI. Interestingly, the mitochondrial kinase that we call CKMT2 did not follow this pattern. Moreover, our in vitro data clearly show that CKB depletion, here is CKB, enhance lipid accumulation in differentiated adipocyte. Here the lipids are labeled with the body P that bind to lipids, to neutral lipid, indicating that creatine metabolism can play a regulatory role in adipogenesis. So to study the role of creatine metabolism in adipogenesis, we start to map the expression of every enzymes involved in creatine metabolisms across differentiation from day 0 to day 12 after the induction of differentiation. And only 2 genes, CKB in green and CKMT2 in pink, rose dramatically during adipogenesis. Their protein level also steadily increased during the differentiation, day 3, day 7, day 10. You can see here CKB and CKMT2. An analysis of human biopsies confirms that mature adipocytes, here in green, express far higher level of this enzyme than pre-adipocyte, here in purple. To test the functional importance of CKB and CKMT2, we silence both genes one day before now inducing differentiation. Despite robust knockdown, as you can see in these panels, here it's the expression of CKB. In green, you have the expression of CKB. Here you have the expression of CKMT2. And in purple, you have the expression in the SC-CKMT2 cells. We can see that the knockdown of CKB and CKMT2 do not alter adipogenesis gene program, because PPAR gamma activity, as you can see here, the gene target of PPAR gamma is not altered during the differentiation after SC-CKB and SC-CKMT2. The adiponectin secretion is not altered, and structural markers was not altered by the scRNA against CKB and or CKMT2. However, it was obvious that CKB depletion massively increased lipid storage, but do not disturb the adipogenic gene program. You can see that body P intensity rose, lipid droplets are much bigger than SC control and SC-CKMT2. And there is much more triglyceride in the adipocyte after CKB depletion. Conversely, in CKMT2 depleted cells, there is no this effect. There is not an increase of lipid accumulation. This lipid accumulation is truly observed after CKB silencing. To understand why lipids accumulated, we did an RNA sequencing on SC-CKB versus SC-CKMT2 and versus SC control. we observe that RNA sequencing analysis shows that CKB depletion activates the de novo lipogenesis program. DNL is a pathway by which adipocytes synthesize fatty acid from glucose, especially when carbohydrate availability is high. Key enzymes include ATP citrate lyase, ACLY, acetyl-CoA carboxylase, ACC, and fatty acid synthetase. In SC-CKB, all of these enzymes were strongly induced, as you can see in this here in this heat map. This reflected a true metabolic rewiring. The cells was converting more glucose into fat after the silencing of CKB. Using tricyclic glucose incorporation assay, we confirm that SC-CKB adipocytes show markedly elevated insulin-stimulated de novo lipogenesis, matching with the transcriptional profile. This was confirmed in vivo because in adipocyte-specific CKB knockout mice, we observed a strong induction of DNL genes expression in the inguinal white adipose tissue, FASN, GLUT4, ACC, SCD, or ACLY. And this was associated with an increase of the other master regulator of de novo lipogenesis, SRBP1, which is strongly induced here, as you can see here, in the CKB knockout mice in inguinal white adipose tissue. Thus, CKB represses lipogenesis in vivo as well as in vitro. Conversely, CKB overexpression in human pre-adipocyte before differentiation leads to a reduction of DNL gene expression and a reduction of lipid accumulation in the adipocyte. Again, CKB overexpression do not disturb the expression of pan-adipocyte marker. This data suggests that CKB function as a metabolic brake on-off lipogenesis. Because glucose fuels DNL, we examine glycolysis by sears. SC-CKB here in green show higher extracellular acidification, meaning higher glycolysis. And this increase of glycolysis activity in SC-CKB cells was associated with an accumulation of sugar intermediates, such as fructose 6-phosphate, pyruvate, and xylulose 5-phosphate, and also lactate levels in the comparison in black SC control versus SC-CKB. These metabolites directly feed the lipogenic machinery, connecting CKB loss, increased glycolysis, and increased de novo lipogenesis. This elevation in sugar phosphate, xylulose 5-phosphate and glucose 6-phosphate, point toward the key transcriptional regulator, SHREB. SHREB is a glucose-activating transcription factor that turns excess carbohydrate into fat by driving DNL gene expression, de novo lipogenesis gene expression. When glucose metabolites rise, SHREB become activated and promote the expression of ACLY, ACC, FASN, or SCD1. Indeed, SC-CKB adipocytes displayed a strong induction of the active form of SHREB at the site of 35 kilodalton. In SC-CKB, if you compare, there is a strong induction of the expression of SHREB in the nucleus. This confirms the link with the glycolysis. And one thing was also very interesting, it's when we inhibit glycolysis by treating the cell with 2DG, we normalize SHREB level. This confirms a link where more glycolysis induces SHREB activity that promote de novo lipogenic genes expression. To test the necessity, we silence MLX high PL the gene encoding Sheb, and this completely abolishes Sheb protein expression, as you can see here. And this also reduce DNA gene expression induced by SCCKB, here in purple, if you compare SCCKB and SCCKB plus SC-MNX-IPL. And this also normalize lipid accumulation induced by SCCKB. Thus, Sheb is the essential transcriptional effector downstream of CKB loss. A final question: why does glycolysis increase when CKB disappears? We look upstream and focus on the PI3K AKT pathway, a central upstream regulator of glycolysis and a key mediator of insulin-stimulated glucose metabolism in adipocytes. Indeed, AKT is not only a well-established activator of glycolysis, but also listed as a potential CKB interactor in the NCBI gene datasets. Indeed, as you can see here, immunoprecipitation of AKT in our human adipocyte model confirms a physical association of AKT with CKB. Furthermore, CKB knockdown markedly increase AKT phosphorylation, as you can see here in the Western blot, here in the right, a reliable marker for the AKT protein kinase activity. Using AKT inhibitor that we call MK2206, we erase the entire lipogenic cascade. We find that AKT inhibition first reduces Sheb activation, decrease DNA gene induction, and reduce DNA activity, as you can see here in purple, if you compare SC control and SCCKB and SCCKB treated with NK that inhibit AKT. And this will reduce the lipid accumulation induced by CKB silencing. Together, these results reveal a current mechanisms. CKB normally binds to fine-turn AKT. Thus, loss of CKB release AKT. AKT will be increased. This will increase the glycolysis. The intermediate of glycolysis will increase, and particularly the phosphosugar, such as xylulose 5-phosphate or glucose 6-phosphate. This will increase the activity of Sheb, and Sheb will induce the expression of the lipogenic gene expression. In conclusion, our work uncovered a simple but transformative idea. In obesity, phosphocreatine builds up in white adipocytes, not because adipocytes produce more of it, but because they can no longer use it because CKB is not here. And the reason is clear, because CKB is down-regulating at the epigenetic level with a higher DNA methylation on its promoter. Once CKB disappears, the creatine phosphate shuttle breaks down and which its CKB ability to restrain AKT. AKT loses its brake, it becomes overactivated. AKT overactivation leads to increased glycolysis and accumulation of ATP in adipose tissue cells. So in mature adipocyte, accumulation of phosphocreatine and ATP inhibit AMPK, a kinase that have an anti-inflammatory function that is characterized by a strong secretion of CCL2 by adipocyte, which is a macrophage chemoattractant that drives immune cells infiltration, in particularly macrophage, that is known to promote insulin resistance. And during adipogenesis, the same AKT-driven glycolytic surge fuels Sheb hyperactivation. As a result, early adipocytes ramp up fatty acid synthesis, accumulate triglycerides, and are pushed toward hypertrophy. So thank you very much for your invitation. I would like to warmly acknowledge all the collaborators that made this work possible, especially the Microbiome group and Laurence Kazak lab. I want to also to thank Professor Karine Clément, Geneviève Marcelin, and Emmanuel Gauthier. And I also want to thank my colleague Salon Magdazi, Jean-Luc Ranzi, Gianluca Ranzi, that we called Jean-Luc, and Roman Igos, whose contribution have been essential to this project. And finally, thanks you for bearing with my English. I'm very sorry for my level of English. It's not good, but I'm sorry. And I truly hope this journey into adipocyte was interesting, and I'm very happy to take all of your questions. Thank you so much.
Thank you, Simon. Very nice talk, a really good presentation. I have one question before we start for the others. Thinking about adipocytes, you showed a lot of experiments demonstrating CKB importance in metabolic homeostasis. This is really amazing and really nice history. Do you believe that CKB could be a target to be increased? And if it is the case, thinking about obesity and all of these metabolic homeostasis, how we could do that?
Yeah, this is a very nice question, because this is also for sure in link with our study. It's and the link also, like I said, about the Laurence Kazak works. So one of our idea from this work is to promote the phosphocreatine catabolisms by increase CKB activity. This could have two different advantage. The first advantage is to reduce phosphocreatine accumulation in adipose tissues. That will reduce inflammation according to our work, but also, if you think more broadly, and from the study of Bruce Spiegelman and Laurence Kazak, this will also potentially increase the mitochondrial activity and the thermogenic program, and increase the creatine fatigue cycle. Because a paper from Jan and Rabani have shown that the creatine fatigue cycle is mainly mediated by CKB. So this can have two advantages if you want.
No, really nice. Next question, Carlos. Please, thank you for being here.
Hi, thank you. I'm going to briefly introduce myself, Carlos Escande from Instituto Pasteur Montevideo. Very, very nice talk, Simone. I have read your papers. It's an amazing work that you have been doing. So congratulations for all the work and for the talk. So I have many questions, but I'm gonna ask a couple of one is you mentioned briefly during your introduction that GATM does not express in mature adipocytes. And there are some works. So there are actually Lawrence was one of the authors of one of the papers showing that knockout of GATM in adipose cells led to obesity and cold intolerance and all that. So if what do you think about the role of GATM, or if you have any kind of evidence that actually is actually not playing a role in local creatine synthesis in mature adipocytes? And then I have another question regarding the the downregulation of CKB. If you have any clues about what can be the driver of the silencing of CKB regarding signaling, if it's some metabolic flux that is changing either methylation, or it's just signaling that it for some reason is silencing CKB expression. If you have any clues of how this is how this is happening. Yes. And the final question, and this is maybe I missed a little bit during the introduction, is how do you think the creatine system is working in mature adipocytes? Do you think it's the classical mitochondrial cytosolic shuttling of creatine phosphocreatine, or you think that more than CKB is the one that is running the show, both as a kinase and as a or there is a phosphatase going on. So what are your thoughts about that?
Okay, thank you so much. Thank you so much for your question. Very interesting question. So regarding the synthesis of creatine in the cells, so in our model, and we also confirm that in mice, we find that mature adipocyte. I'll talk about white adipocyte. White adipocyte are not able to synthesize creatine. So the source, so we measure, when you do the experiment, we measure creatine and phosphocreatine in the cells. And we think that the creatine comes from the SVF, but is not produced by the cells because the cells do not express GATM. If we talk about adipose tissue is complex. So you have a lot of cells, and especially immune cells express GATM. So we think that there is a communication between the cells. It's a work with Salom Magdazi. We try to understand how creatine can shuttle between immune cells and adipocytes. So we try to understand that. So for us, it was a challenge to understand what happened. Regarding CKB, so we did, like I said, the transcriptional profiling. So we treat the cells adipocyte with insulin, with catecholamines, with a lot of things. And what we find, it's clear. What downregulates CKB, it's estrase, and specifically the XBP1 branch. And XBP1 is activated after when you have a surge of fatty acid, because in hypertrophic adipocytes, you have an increase of free fatty acid in the cells, and this will lead. It's a paper. It's all the work of Bob Faresi and Toby Walter, and this no esterified fatty acid leads to promote estrase, and estrase via XBP1 will go to the promoter of CKB, and on the promoter of CKB will recruit DNMT to increase the methylation of the promoter of CKB, and this is why CKB downregulated in obese adipocytes. For the last question, so yeah, from our work, because as you have talked about also the work of Laurence Kazak. So Laurence shows an important thing that we not observe in white adipocyte. Laurence shows that CKB is expressed in mitochondria. Usually we find in the mitochondria the CKMT isoform. But Laurence say when you put mice on cold exposure, you increase the expression of CKB directly on CKB. And CKB will work with TNAP to create this creatine futil cycle. So this is well shown by Laurence and by Bruce Spiegelman. And this will increase the thermogenesis. But we can, in mature adipocyte, in white adipocyte, this will not work like this, because CKB is only expressed in cytosol. So when you do silencing of CKB, we deplete the creatine kinase activity in the cytosol, and in the cytosol, the sense of the reaction is to promote the formation of creatine and ATP. And this is not possible when you have not CKB. So you will promote the accumulation of phosphocreatine. So, and this, it was difficult to understand what happened. When we do that, there is a huge increase of mitochondrial activity and glycolysis. And during several years, we tried to find how phosphocreatine accumulation can promote this metabolic rewiring. And we find that CKB can directly bind and work with AKT, like a moon-like function of CKB. And it's an allosteric modulation of CKB to AKT. And CKB, if you want, modulates fine-tune the AKT activity. So when you remove CKB, AKT become overactivated. And this will promote mTOR activity, and mTOR activity is a strong inducer of glycolysis. And this is associated with an increase of mitochondrial dysfunction and stress in white adipocyte and lead to ATP overproduction that lead to NPK inhibition. So, and I hope I respond to your, I answer your question. I don't know.
No, yes. Great answer. Thank you very much for sharing all your thoughts.
Thank you so much for your question.
So, because of the time, I start with the questions. Thank you, Carlos, for the questions, and also thank you, Simon, for your talk.
Thank you so much.
Moving on, I'm delighted to invite Dr. Amanda Carla de Oliveira. Amanda was a very special person for us because she has a connection with our institution, holding a PhD in science with an emphasis in physiology, right here from UFMG. She actually completed her doctoral research internship at Sorbonne Université, which paved the way for her current role because she is now conducting her postdoctoral fellowship there in Paris. Continue her focus on adipose tissue remodeling. Amanda, please go ahead.
Hi, everybody. Thank you. I will share my screen. I'm really happy to be here. This group is really, this is really special to be here, to be presented to you by you, Marina. That was my co-advisor doing my master.
My pleasure.
So, as Marina said, I'm a postdoc. And today I'm going to talk about my ongoing project that is regarding adipose tissue remodeling. That's regarding adipose tissue remodeling during weight variation. The control of the metabolism, the systemic metabolisms, it's something that it's something controlled by several organs, but today I'm going to focus on white adipose tissue. White adipose tissue, just remind you something that Simon already said, it's an endocrine organ that is responsible for functions like glucose and lipid homeostasis, feedback control of food intake, insulin sensitivity, the secretion of adipokines like leptin, adiponectin, energy expenditure, energy storage and release. I won't go in details, but the healthy adipose tissue orchestrates uniquely the systemic metabolism. And under situations of a positive energy balance, adipose tissue can expand, can remodel in a healthy way. And this happens with an increase of adipocytes, which is called adipogenesis, and also by the increase of new blood vessels, the angiogenesis. However, when this remodeling fails, it becomes pathological, with a reduction in adipogenesis and an increase in adipocyte hypertrophy, inflammation with an increase of infiltrations of immune cells like macrophages, and also fibrosis. All this remodeling in adipose tissue, it's called fibroinflammatory remodeling. In our lab, I'll change this. Sorry. Our lab is focused particularly on fibrosis. Fibrosis is defined by excessive production and accumulation of extracellular matrix. Here, you can see a human visceral white adipose tissue histological section is stained with red picrocereus, and here is under white light. Okay. Here is the same image now under polarized light, and you can see what serious red stains, that is collagen fibers. And fibrosis, the fibrosis that happens in adipose tissue during obesity is closely related to now worse in a function of adipose tissue. And this is characterized by unhealthy adipocyte hypertrophy, as Simon already previously explained, inflammation, inhibition of adipogenesis, and glucose intolerance. All this impairs the function of adipocytes and leads to a multi-organ failure. And this I highlighted for you, for example, insulin resistance in liver by the deposition of fat in liver, insulin secretion defects by the pancreas, and also insulin resistance in the muscle. And this multi-organ failure is related to the increase of comorbidities like type 2 diabetes. So, because of this, it huge the search of strategies to treat obesity and also the comorbidities. And the main strategy that for obesity rely on weight loss. Indeed, weight loss of 10% induce a metabolic improvements like in reduced insulin resistance, reduced dyslipidemia, reduced hepatic steatosis, and improvement in sleep apnea. However, sustained weight loss remains a major challenge for those patients. And here I make a question for you. How about adipose tissue in weight loss? Our lab long ago started to investigate this, and they used it as a strategy, weight loss induced, bariatric surgery induced weight loss. And they collected biopsies from adipose tissue before the surgery, 3 months and 12 months after the bariatric surgery. And as you can see here, there is an increase of collagen deposition after the bariatric surgery. So it seems that there is no fully rescue of adipose tissue after weight loss. And in agreement with this data, I bring to you recent publication based on large transcriptomic data showing that adipose tissue after weight loss keeps some features of obesity. So, since the adipose tissue do not recover after weight loss, I ask to you, what are the consequences for weight regain? And to address this question, we set up a mouse model of weight cycling, or WC. We used C38 mice that are more prone to develop fibrosis in epididymal adipose tissue upon high-fat feeding, which is more similar to what happened in adipose tissue in human during obesity. So we took those mice and first we did a diet-induced obesity with high-fat feeding for 4 weeks, then followed by a shift to show diet to induce weight loss. And then in the last 4 weeks of the experimental period, the weight cycling mice were then compared to obese mice that were exposed to just one cycle of obesity. And as you can see here in the body weight and in the fat mass, at 12 weeks, the both groups are similar. However, when we perform the glucose tolerance here in the dashed line in blue, you can see that the weight cycling group is glucose intolerant. And this is accompanied by the increased home IR index. So then we decided to analyze the fibrosis deposition. We decided to analyze the visceral adipose tissue, here represented by the epididymal adipose tissue. And as you can see here in this image stained with Sirius red, there is an increase in fibrosis deposition in weight cycling group compared to obese mice. We then asked about the function of adipose tissue. And for this, first, we analyzed the expression, the mRNA expression of molecules that are responsible for the characteristic of adipocytes. Those molecules are adipokines like leptin, adiponectin, the lipases, and also perilipin. And as you can see here, the weight cycling downregulates the expression of all those enzymes. So moving on, we decided to evaluate the insulin sensitivity by assessing the phosphorylation of AKT by the stimulus of insulin. And as you can see here, the weight cycling group showed a reduced phosphorylation of AKT upon the stimulus of insulin, suggesting an insulin resistance in this tissue. So collectively, our data suggests that despite the same body composition between obese and weight cycling mice showed a metabolic deterioration with glucose intolerance, epididymal adipose tissue insulin resistance, and epididymal adipose tissue fibrosis. This also suggests that maybe the weight loss can induce some dysfunction in adipose tissue. So next, we decided to investigate the effects of weight loss on the structure and the cellular integrity of visceral white adipose tissue. And for this, our experimental groups, we developed a protocol to induce weight loss, and in our experimental groups were lean mice fed the entire period with just show diet, obese mice fed for 4 weeks with high-fat diet, which is the mice that were before the induction of weight loss, and formerly obese mice that are obese mice, followed by a shift to shown diet to induce weight loss. And as you can see here in the body composition, the weight loss indeed restore at the lean level the body composition. And as expected, before the weight loss in the obese mice, we observed a glucose intolerance in these mice. But this is normalized after weight loss, as we can see the overlap of the lines between formerly obese mice and lean animals. We then start to ask it also about the function of adipose tissue. And for this, we performed a lipolysis experiment, which consists to use explants of epididymal adipose tissue, stimulated with the selective agonist of the adrenal receptor beta 3, CL316, and then we analyzed the glycerol release. So, in basal condition, obese mice and formerly obese mice showed an increased glycerol release compared to lean animals. Under the lipolytic stimuli, we observed just the lean group showed an increase in glycerol release. And also, we observed that this outer response to lipolysis is showing in the expression of lipolysis-related molecules, here in the MRR level, but also in the proteomic level. We also evaluated the staining of perilipin, which is a protein that surrounds the lipid droplet in the white adipocytes and is also a marker for adipocyte characteristic. And as you can see here, in the formerly obese mice, there is an increase of perilipin negative adipocytes compared to lean animal, suggesting a loss of adipocyte function. This data and the lipolysis data suggest this loss of adipocyte function in the weight loss. Sorry for the ambulance. And since fibrosis is related to a reduction function in adipose tissue, we evaluated the fibrosis deposition, and as in human, there is an increase of fibrosis deposition and weight loss. And since fibrosis is instrumental for the reduction in the adipose tissue functionality, our next question was to understand the mechanism underlying why this fibrosis increased during weight loss. And for this, we took our formerly obese mice, and then we compared to obese mice that were fed with high-fat diet for 8 weeks. And those mice, when we compared in body weight, fat mass, epididymal adipose tissue mass, and the mean adipocyte area of this depot, we observed an increase of those parameters in obese mice compared to formerly obese mice, showing that phenotypically they are different. And to go deeper in this difference, in this phenotypic difference, we performed a proteomic analysis in the epididymal adipose tissue, and we identified several proteins that are differentially regulated. And among the proteins that were downregulated in formerly obese mice, the top one, it was the piperacillin pathway, suggesting a dysfunctional adipose tissue. And in agreement with this, we observed that the top one upregulated pathway, it was the oxidative stress and redox pathway. So weight loss seems to be a stronger inducer of white adipose tissue dysfunction and stress compared to a obesogenic condition. However, despite these adipose tissue and those animals being different, their fibrosis is quite similar, as you can see here in the serous head staining and also in the hydroxyproline content. We next analyzed the composition of extracellular matrix, and we performed a dedicated proteomic analysis for ACM extramatrix cellular, and we identified 158 proteins. And what we observed that there is no difference between the groups. So it seems that weight loss-induced fibrosis and obesity-induced fibrosis are quite similar. And since the fibrosis between weight loss and obesity are similar, we asked if pharmacological strategies that work to treat fibrosis during obesity could work during weight loss. And we recently published a paper using an antifibrotic and anti-inflammatory cocktail where we administered during the high-fat feeding this drug, and we observed that there is a reduction in fibrosis. However, when we administered during weight loss this drug, we observed no difference in fibrosis. So this suggests that the mechanism that drive the fibrosis in weight loss are different from obesity. Sorry. We then decided to investigate the mechanism behind. And our lab, more precisely Geneviève Marcelin, published that demonstrated that progenitor cells that express under their surface the CD9 marker, are the main responsible for the secretion deposition of fibrosis in epididymal adipose tissue during obesity. The proliferation and activation of those progenitor cells is dependent of molecules secreted by macrophages. So our first question, it was, how about the macrophages during weight loss? So for this, we've performed first a flow cytometry analysis, and we observed that, as expected, in obese animals, there is an increase in macrophages. And in weight loss, we observed that those macrophages remain in epididymal adipose tissue. We next asked if these cells remain in the epididymal adipose tissue, what are their profile? And for this, we performed a cell sorting for those macrophages, and we phenotyping them with RNA sequencing. And what we observed, it was surprised because the top one downregulated pathway, it was TNF signaling. So this suggests that despite these macrophages remaining adipose tissue after weight loss, they show a reduced pro-inflammatory activity. And this is also suggests a reason to why our antifibrotic anti-inflammatory cocktail didn't work to reduce the fibrosis induced by weight loss. Then we focus our attention into the progenitor cells, that are the cells that secrete, as I said, the cells are responsible for the secretion deposition of fibrosis in obesity. We first performed a flow cytometry, and we observed that the progenitors are increased in obesity and they remain after weight loss. Next, we analyzed the proliferation of those cells, and what we observed that in obesity, as expected, there is an increase of proliferation, there is an increase of Ki67 compared to animals, and in weight loss, there is a reduction of the proliferation of those cells, suggesting that these progenitor cells, they have a deactivated profile. They have a deactivated characteristic. We then asked about the profile of those cells, so we also performed the flow cytometry, cytometry cell sorting, and we performed the RNA sequencing in those progenitor cells, and we analyzed the EMT genes that are related to fibrogenic activation in progenitors. We took those genes that are upregulated in obese progenitor cells compared to lean. And what we observed when we analyzed in formerly obese progenitors from epididymal adipose tissue, that there is a downregulation of the fibrogenic genes. In our data, in the whole adipose tissue, in the whole epididymal adipose tissue, is in agreement with this data from the progenitor cells. Because when we analyze the mRNA expression for fibrosis components like collagen, fibronectin, and CTGF, we observed that after weight loss, there is a reduction in the expression of those genes. So it seems that there is this suggests that there is a reduction in the synthesis of fibrosis components in adipose tissue after weight loss. But I would like to remind you that fibrosis is driven by a balance by production and degradation, and in obesity there is an increase in the production of the fibrosis, which leads to the increase of fibrosis deposition. In weight loss, we observed that there is a reduction in the fibrosis synthesis. So our question focuses in if maybe there is a reduction in the degradation of collagen. And here I would like to talk quickly with you about collagen degradation, which is a complex process that starts in the extracellular compartment with the action of proteins like metalloproteinases. Those these collagen fragments, they are internalized by transporters, and then the digestion is finished in the lysosomes. And to evaluate this collagen degradation, we set up an experiment, an ex vivo experiment with called the Cube, which is a probe that is, when it is not is undegraded, is the molecules that are fits, they are conjugated, so they do not emit fluorescence. But when they are incubated at 37 degrees, and there is the presence of degraded enzymes, they emit deficit. So the cells that are collagen degraded positives, they emit FITC by the fluorescence. And we measure this by flow cytometry. In our first question, it was about what cells in epididymal adipose tissue are the responsible for the collagenolysis. So for this, we incubate the cells at 4 degrees, a temperature that the probe, the cube, does not work. And also at 37 degrees, the temperature that the cube works. And as you can see here, you observed a shift, not in the endothelial cells, but I shift in the progenitors and I shift in the macrophages. And here in the quantification, you observe that the main cells responsible for collagenolysis are the macrophages and the progenitors. So then we analyzed the Q in our experimental group, and what we observed that in macrophages and in progenitors, there is a reduction in collagen degradation in obese and in formerly obese mice. So as a conclusion, I bring it to you that weight loss indeed improves systemic metabolism, like insulin resistance. But in epididymal adipose tissue, it seems that is not fully recovered because the function of adipose tissue is still not good, not working, and also there is an increase of fibrosis at the same level as obese mice. However, despite the fibrosis be similar between obese and weight loss mice, the mechanisms are different, as evidenced by the failure to the antifibrotic cocktail in formerly obese mice. So our suggestion is what I'm currently working right now in my postdoc is about drugs that focus on the mechanism that drive the collagen degradation and reset the function of epididymal adipose tissue. And here I have a lot of people to thank. I thank my current lab, Nutriomix, especially Professor Karine Clément, my boss, Geneviève Marcelin, the Clémentine Rébière, a former PhD student that performed part of this experiment, the rest of my group. I also special thanks to Immunometabolism group that we had this partnership that I did part of my PhD as a immunometabolism student here, and now I'm from Nutriomic. Special thanks to Adaliene, to Marina. A special thanks to Agatha and Adma, the students that were here with me and they helped me in this experiment, and the past students too that also helped. And of course, thanks to the financial support, and thanks to you that are watching me.
Thanks, Amanda, very much for your really, really nice talk. It was amazing, and I will do the first question just about this end part related to fibrosis, that the obese mice and the formerly mice,
formerly obese mice,
obese mice, okay, that they seem to present a different response in relation to medicaments. Is it possible that the type of formation of fibrosis is different in the sense that for example, you have in obese one type of formation of
fibrosis.
It is possible to measure these because it seems so it's not strange, but it seems that they are completely different, and that maybe something related to the formation is different, or the way that they are put together, you know.
I will explain to you. So here, I don't know, my screen is still sharing. How to end this sharing of the screen? I'm lost.
It's an open part. It appears something if you go up. I believe that you
here.
Yeah, more.
Okay, no, but it's fine. I will explain.
Maybe I can.
Okay, so here I show this data because what we performed here, this data here that I'm surrounding, is the analysis of the matrizome. What is the matrizome? Matrizome is the proteins that compose the extracellular matrix, which includes the components of extracellular matrix like collagens, fibronectin, elastin, and also the proteins, the enzymes, and the cytokines that regulate these extracellular matrix. So indeed, here I didn't show the image complete, but indeed we analyzed the composition of the extracellular matrix between mice, obese mice for 8 weeks of high fat and the formerly obese animals. And what we observed that the matrizome of these two groups, it's really similar. So it seems really that the composition of the fibrosis between these two groups is similar. So it's not exactly a matter of composition.
Something more that maybe it is there that is working against this part of
Yes,
the fibrous
yes,
degradation.
yes. Okay, how I finish? Okay, fine. Sorry.
Okay. The next question is from Gabriela Araújo. Amanda, congratulations on your presentation. It was amazing. The weight cycling model is very interesting. I'd like to know if you already evaluated the response of subcutaneous adipose tissue in this protocol.
Yes, we evaluated the response of subcutaneous weight cycling experiment, and indeed, what we observed that this defect in what we observed in weight cycling is just in epididymal adipose tissue. The subcutaneous adipose tissue, when you compare obese and weight cycling, there is no difference between the two groups. The increased effect of weight cycling compared to obese is observed only in epididymal adipose tissue.
Okay. I believe that we don't have more questions.
Okay.
So
Thank
we'll
you.
move forward. Thank you, Amanda. Now we transition to our short talk session, showcasing rising talents from our own immunometabolism research group. First, I'd like to call Adma Babêtto. Adma holds a master's degree in science focused on physiology and is currently a PhD candidate in physiology at UFMG. She has been doing interesting work within our group. So Adma, please share your slides whenever you're ready.
Can you hear me?
Yeah.
And can you see my shared screen?
Yeah, it's not on the white screen, but I can see your screen.
Okay. Let me
put on
just,
presentation.
oops, put the pointer. So thank you, Marina, for the introduction. I will try to be brief because I have
not see the total screen, the presentation.
Ah, you can see?
No, not yet.
No?
It's just
Let me try again. Stop sharing.
Maybe share the window.
And now it's full?
No, not yet.
Not yet?
No.
Let me try again. Stop sharing. Share the screen. And now?
Yeah, now it's working. So good.
No?
Yes. No, it's good. Can start.
Ah, it's good. Okay, so I'm sorry for that. I'll try to be brief because I only have 10 minutes to talk. As Marina said, I'm a PhD candidate and my advisor is Adeliene. And today I will be talking about my research topic, which is adipose tissue expansion as a driver of cellular senescence in obesity. Let me just, where is the pointer? So cellular senescence is a stress response in which damaged cells permanently stop dividing. And this process is primarily controlled by two pathways, the P16 pathway and the P21 pathway, which is a cell cycle arrest pathway. And they will be responsible to prevent the damaged cells from proliferating. And one thing that is very important here is that the senescent cells will undergo structural changes and functional changes and become very resistant to apoptosis. One other thing that those senescent cells will present are the senescence-associated secretory phenotype, which are cytokines, chemokines, growth factors that will be responsible to recruit the immune cells for clearance and will be responsible for the communication between the senescent cells and non-senescent cells between tissues or in the tissue itself. So they are responsible for the enhancing fibrosis, inflammation, regeneration and repair, and proliferation. So these CESPs that I was talking about, they are signals that are powerful drivers of chronic low-grade inflammation, which is a hallmark in aging and in obesity. And those senescent cells will present a chronic inflammation and insulin resistance that we can see clearly in obesity as well. So one thing that was raised, one question in this field was, could cellular senescence represent the mechanistic link between obesity and metabolic dysfunction? We already have this answer because in this article of 2009 from Minamino, they show that in overall, obesity-driven tissue expansion, and this tissue expansion promotes accumulation of senescent cells through those triggers that I said in the beginning of my presentation, like oxidative stress, DNA damage, and telomere dysfunction. And in aging, we already know that the adipose tissue shows a clearly accumulation of senescent cells that can communicate through the CESPs. So the CESPs play an important role here. And we don't know yet if in the context of obesity, the same thing happened in adipose tissue. So another question that we have is that could adipose tissue senescence represent a starting point for metabolic dysfunction and discommunication with the other organs? We have some studies in mice that have shown that removing the cellular senescence can improve the tissue function and can extend the life of those animals, suggesting that these cells are important and active drivers of age-related dysfunction. So this highlights the importance of targeted senescence, since several strategies exist in the literature targeting those cells like NAD boosters, senolytics, metformin, but one strategy called our attention of our group, which is if dietary treatment alone can possibly reduce these senescent cells. So, as I said, it is still not completely clear what the temporal dynamics of cellular senescence in obesity, if there is a tissue that presents an enhance in those cells in a premature way, and whether dietary intervention alone is capable of reversing senescence markers in metabolic organs. So to assess that, we did a temporal evaluation of cellular senescence, and we used black 6 mice, male mice with 8 weeks old, and we divided them in a group that received a chow diet and one that received a high-fat 45% diet. And we evaluated this diet until 16 weeks, and we collected tissue in all of those time points to understand where there is an enhancing cellular senescence. So the first thing we did was to evaluate the SA beta-gal, which is a lysosomal enzyme, and this enzyme tends to accumulate in senescence. And as you can see here in the epididymal adipose tissue, you have an increase in this SA beta-gal with 8 weeks of diet, and this enhance tends to continues until 16 weeks. And the same thing happened here in the inguinal adipose tissue, where we have increase in this lysosomal enzyme as well. We evaluated in liver and gastrocnemius, and there was no different gastrocnemius muscle, and there was no difference in those tissues between the weeks of diet. The second thing that we did was to evaluate the expression of the cell cycle REST genes that are very important in those senescent cells in both adipose depots, epididymal and inguinal. And regarding the P53, there was an enhance only with 16 weeks. And when we evaluated the P21 in the epididymal, it was an enhance in the expression of this gene with 4 weeks, and this expression continues. When we look at the inguinal adipose tissue, there was a difference only in the P21. So seems like this cell cycle arrest gene is important in both of the tissues. We evaluated as well in the liver, and in all of them, there was an enhanced only with the 16 weeks of diet. So there is a difference in those parameters and between those organs when we talk about the expansion of the adipose tissue and the obesity. We evaluated as well the intensity of fluorescence of the laminin B1 marker, which is a nuclear membrane marker, and there was a decrease only in the inguinal adipose tissue of the intensity of fluorescence of this marker, which is a clear hallmark of senescence with 8 weeks and 16 weeks of diet when compared to those control mice. Next, we evaluated a DNA damage marker, which is H2AX, which is a very important marker of senescence. And again, on the epididymal adipose tissue, we have the presence of this marker before the inguinal adipose tissue, which we have a presence of this marker only with 16 weeks of diet. So there is a difference between the tissues and the depots as well. Lastly, we evaluated those tests that I said at the beginning of my presentation in both of the adipose tissue depots. And regarding the inflammation, we have an enhanced in CCL2 and SPP1 in the epididymal, and the same thing happened in all the other fibrosis and remodeling markers that we evaluated on this tissue. When we look at the inguinal adipose tissue, we almost had the same results, but only SPP1, there was no difference between the weeks. And we can see again that 16 weeks plays an important role here because it seems like every difference and every reorganization of those cells is occurring within those weeks of diet. We did the same evaluation on the liver, and there was no difference on the inflammatory genes, but there was enhanced in fibrosis and remodeling genes. So in summary here, at first we were trying to look if there was a difference between the depots and the tissues, the organs itself. And we could see that the epididymal adipose tissue appears to have unhealthy cells with P21 high cells, a lot of SAS factors, MMP2, TIMP1, TIMP2, inflammatory factors are present, and there are the presence of SR beta gal as well. And inguinal adipose tissue, it's a little bit similar, but then that the start here of this process of cellular senescence seems there is the epididymal adipose tissue and not the inguinal, since we have a lot of alterations first in the epididymal when compared to the inguinal. And when evaluated the liver, there was not much of alterations with this week of diet, 16 weeks, only some P21 high and P53 is 16 as well, and the presence of some SAS factors. So after that, we asked ourselves if are the tissue senescence changes induced by high-fat diet reversible with dietary intervention, and do different interventions have similar effects? So to evaluate that, we did the same 16-week obesity induction treatment, and after that, we divided the obese group in two other groups, which were the groups of the dietary intervention. One group underwent a 24-hour intermittent fasting, so they were 24-hour fed and 24-hour fasting for 10 weeks. And the other group had a switch of the diet, so they started to receive a chow diet here for 10 weeks as well. So here in this treatment, the group that was that received the switch of the diet had a decrease on the mean caloric intake in the beginning of the treatment. And regarding the body weight, there was a decrease in both groups, the intermittent fasting and the switch of the diet. And when we did the delta weight loss, the group that received the change of the diet had a decrease in the delta weight loss. So when we look at the intermittent fast, some animals didn't lose any weight at all. And as expected, those animals that had the switch of the diet had a decrease in epididymal, inguinal, and leptin. And the intermittent fast, nothing happened with this group. When we look at the SA beta-gal, in the epididymal, they did not show a difference on the obese that had the shift of the diet, the switch of the diet, but they seem to have a tendency here. And when we look at the inguinal adipose tissue, they completely reverse the signal of the SA beta-gal when compared to the obese ones. We look at those cell cycle rest proteins, and after the intervention, only the P21 was decreasing the epididymal. And in the inguinal, we tend to have increasing in the intermittent fasting group and a decrease in P21 as well. So it seems to be an important cell cycle rest gene here, the P21, in both adipose depots. And when you look at the liver, only the P21 on the group that received the chow diet have a decrease in this gene expression. So in summary of this project, we want to look at other things, but in the group that received the intermittent fasting protocol, we have the presence of the marker of SA beta-gal in both tissues. And when we compare to the group that received the chow diet, we have a decrease in the inguinal, in P21 and beta-gal as well. And when we look at the epididymal adipose tissue, we have a decrease in the P21. So seems like we have more reverse of these alterations that is caused on the senescent cells in the group that received the chow diet. So seems like it's very important the type of nutrient that those mice are receiving. And we want to do a lot of things right now. We want to check some inflammatory factors in this experimental design, some SASP factors, the lambin B1, and we want to check as well which are the type of cells that are involved in the senescence of the adipose tissue, because we really don't know right now. We have a hint, but we want to look further in this subject. So I want to thank you all. Thank you, my research group, and all the other groups, the nutriomics that received me this last year, and all the funding agencies.
Thank you, Adma, for your amazing presentation. We will go directly. Carlos, you can ask a question.
Oh no, I can ask later if you
No,
prefer.
you can. You can.
Okay.
No,
okay,
you can.
Okay. Very nice talk. Thanks. So I'm very, very curious about what do you have any clue about what is happening to those senescent cells that disappear during the treatment? Are they being cleared? What happens? Do you know any speculation?
You were saying about the dietary treatment?
Yeah.
We don't know yet because I need to do these experiments, but I think probably on the group that received the chow diet will be a decrease on those SASP factors. This is my hint, because we don't see any difference in tissue weight in terms of intermittent fasting, only the one that received the chow diet.
Okay. Thanks.
You're welcome.
Okay, so we'll go directly to our next short talk. Next up, I'd like to invite Julia Magalhães. Julia is also a member of the Immunometabolism Research Group. She brings a background with master's degree in science with an emphasis in physiology. She is currently a PhD candidate in physiology at UFMG. We are eager to hear what you have been working on. Julia, the floor is yours. I still cannot hear you. Oh.
No?
No, yeah, it's okay.
Yes, okay. I'm gonna share my screen. 1 second. Can you see it? It's good?
Yeah, good. Can start.
Okay, perfect. So I'm gonna present, and my name is Julia, and I'm gonna present some preliminary data that we have on a real-life setting protocol to assess metabolic flexibility. So we are working with the concept of metabolic flexibility, which is about the organism's or the tissue or the cell's ability to quickly switch between different energy substrates according to their availability. This means, for example, when we are in a fasted state, like an overnight fasting, we have a low disponibility of glucose, and therefore the organism oxidize fatty acids. And when we move to a fed state, there is an increase in glucose availability, and therefore the organism switches to use glucose as energy substrate. This is assessed in general through the respiratory quotient, which is the ratio between the carbon dioxide liberated and the oxygen consumed in the process of oxidation. This quotient varies from 0.7 to 1. As it approaches 1, it means that there is more carbohydrate being oxidized. And as it approaches 0.7, it means that there is more fats being oxidized. And values in between means mixed nutrients. This concept has raised questions, not questions, but has been proposed to sometimes be impaired in some conditions, such as obesity and diabetes, for example. And so it's been said that there is a lower variation in these cases of this respiratory quotients, and therefore a lower change in substrate in these individuals. In humans, it can be systemically evaluated through indirect calorimetry, measuring the respiratory gas exchange of respiration. It enables both to calculate the respiratory quotient and the metabolic rate of individuals. Central in the process of metabolic flexibility and changes in substrate use, there is a dynamical response in mitochondria being proposed in literature, in the sense that in cases of high glucose availability, there is a more fragmented state of mitochondria obtained through a process of fission. And when there is a higher availability of fatty acids, they assume a more elongated conformation and after a process called fission. Also, it's been proposed that there is importance of membranes, intracellular membranes, such as endoplasmic reticulum, sarcoplasmic reticulum, and muscles, where in high glucose availability, these membranes distance themselves from mitochondria. And when there is fatty acid availability and its oxidation, there is an approximation of these membranes. In 2021, Castro Sepúlveda and colleagues have demonstrated that these mitochondrial dynamics can be observed in peripheral blood mononuclear cells, PBMCs, which are easily obtained in humans, which is very important generally in studies, because sometimes it's hard to get biopsies of specific tissues. And what they showed is that after a 16-hour fasting in males, the mitochondria has a more elongated state, while after a glucose overload, it becomes more rounded and maybe fragmented. They've also shown that there might be some differences in the distance from intracellular membranes. The metabolic flexibility has been studied in different settings, very often in conditions that are super physiological, such as clamping glucose or insulin, mostly having hyperinsulinemic clamping, or glucose overload, like the previous work that I've showed. But there has also been works of meal challenges with different compositions, high carbohydrate, high fats, or mixed meals. A lot of times also with liquid meals, trying to evaluate this metabolic flexibility at a more physiological context. So this is an initial phase study where we aim to establish a protocol to assess metabolic flexibility after an overnight fasting and a real-life mixed nutrient meal in apparently healthy lean women. We have data on 15 women aged from 20 to 45 years. They arrive at our lab after an 11-hour of overnight fasting, and after 30 minutes of resting, we measure their indirect calorimetry to determine their resting metabolic rate. Right after, we calculate a meal with common ingredients like industrialized juice, bread, and cheese, corresponding to 20% of their energy requirements, calculated at the time with the resting metabolic rate that we measure, considering their activity, physical activity through metabolic equivalence of activity. And we add 10% of the thermic effect of food. And through 4 hours, starting with a 30-minute measure and then hourly for 4 hours, we repeat the indirect calorimetry exam to capture changes in both metabolic rate and respiratory quotient. We also measure capillary glycemia, and we also have 3 collections of blood samples at before meal, right after meal within 30 minutes of meal, and after 4 hours of meal. And we also apply a visual analogical scale to measure appetite and hunger. So what we have seen so far is that after 30 minutes of meal ingestion, there is a peak in capillary glycemia with an increase of about 43% of the glycemia measured at this time. And then there is a progressive decrease over time. We also have seen an increase in metabolic rate of about 18% after 30 minutes of meal ingestion, and it remains higher than baseline until 4 hours after meal, remaining with about 11% elevation. And respiratory quotient, although it peaked at T30, we didn't see a significant difference in this peak, but it starts decreasing over time, mostly after 2 hours of meal ingestion, and it reaches the lowest levels at 4 hours after meal. We also have seen that this protocol is able to promote satiety 30 minutes after meal and decrease at the same time hunger and desire to eat. Through the blood samples, we were able to successfully separate through fecal gradient PBMC cells and take photographies with good quality. So being able to see nucleus and organelle distribution, for example, in the cells. And we were able to, at different magnifications, observe structures of mitochondria and also intracell membranes. Hopefully, this will enable us to understand if we can evaluate mitochondrial dynamics using this protocol. So the proposed protocol with a real-life setting of meal consumption indicates, has an indication of potential to assess metabolic flexibility in lean women. As soon as we have this protocol well established, we can go further in our research to determine if it can be used in conditions that are proposed to have some impairment in this metabolic flexibility, such as obesity, diabetes, and maybe aging. I would like to thank our research group, especially the researchers that are directly involved in this research, our fomenting agencies, and thank you all for the attention.
Thank you, Julia, for your nice talk. Till now, we'll take a short 10-minute screen break. Please take this moment to grab some water and stretch, and we'll enjoy at 3 and yeah, almost 55. So for our second half of our afternoon section. So see you soon. So welcome back, everyone. To start the second half, we are honored to have Dr. Camila Martinez Clément. Camila joined us with a dual affiliation as an assistant researcher at the University of Buenos Aires and CONICET, and as an honorary associate researcher at the Institute Pasteur de Montevideo. She holds her PhD in biological science from University of Buenos Aires, and her career includes significant international experience, having completed her postdoctoral training and served as a research specialist at the University of Massachusetts Medical School. Camila, thank you for joining us today. We're looking forward for your presentation.
Thank you, Marina, for the introduction. I left you with all the difficult words to pronounce. Sorry about that. So I'm going to start sharing my screen, but fair warning, if my internet is a little bit unstable, if it glitches, I'm going to have to turn off my video, but I will switch it off for the questions.
It's very okay.
Okay. Okay. So today we've been hearing a lot of, first, thank you for the opportunity of presenting my work. And secondly, through the whole day of these wonderful talks, we've been hearing a lot of interesting discussion about particularly white adipose tissue, specifically. So I'm going to try to change that a little bit, and we're going to give a little bit of love to brown adipose tissue. And my talk is going to be divided in two sections. In the first part, I'm going to show you the description of the model that I've been working on, which is the implementation of an intermittent cold experimental exposure protocol. And in the second part, I'm going to show you some data that suggests the possibility of the therapeutical application of this model. Okay, so let's see if I can. So adipose tissue, as I said, comes in at least 3 flavors. You have the white, the brown, and the beige adipose tissue. And besides the morphological differences that you can see here regarding lipid droplet size, the amount of mitochondria, they have pretty much very different function and activities. Also, the distribution of the adipose tissue varies quite a bit, as you can see here in the lower picture of the mice. And there we go. Also, the ability of the capacity of this tissue to, let's say, switch or differentiate into different types of tissue. For example, white can turn into beige, or brown can go into sort of a whitish structure. Also varies depending on the adipose tissue that we are studying. Here you can see, for example, that brown adipose tissue in the mice mainly locates in the interscapular location, and you can see here by the thermal picture that it's basically where you can see very clearly the heat emission. Okay, and as I said before, even though we usually tend to classify, or we want to classify adipose tissue in 3 clear different types of adipose tissue: brown, beige, and white. Not all beige, not all brown, and not all white adipose tissue is actually equal. And you can see it here by a picture that basically shows in a very simplistic way what are the different drivers that could lead to brown or beige adipose tissue. And you can see that it's very complex, and there are not basically one way to achieve a brown adipose tissue or a beige adipose tissue. This basically reflects the complexity of the tissue and the possibilities on how they can switch. Regarding continuing on the adipose tissue, you can also see by this picture that it's a very dynamic tissue. We've seen through the talks again that regarding diet, or here, for example, you can see under different temperature conditions that not only brown fat morphology changes, as you can see, by lowering the temperature, it reduces the lipid content and it gets more active, but also inguinal and perigonal white fat also changes dramatically their structure. In mice, there is a thermoneutral or thermoneutrality zone that is basically beyond, like, around 30 Celsius, which is not exactly the normal animal facility condition temperature that we usually have. We usually have it around 23, and at 23 there is an activation of thermogenesis. So our mice at 33, they are basically doing some basal thermogenesis. Also, the thermogenic capacity of the tissue basically leans on two parts. One is the activation itself of the tissue, that it's basically driven by norepinephrine and a beta-adrenergic activation. We've seen, I think Simone explained the 2 main thermogenic pathways, the UCP1 dependent and the creatine dependent. But beyond the activation, which is an acute response, there's also differentiation that could also lead to an increase in the thermogenic capacity. This differentiation in brown adipose tissue, it's driven by different metabolic gene drivers. Such the canonical ones are PMD16, CBP, PPR, and also the activation of the brown adipose tissue is led by PGC1 alpha. Okay, so let's go to the model that we're working on. The model that we're working on is an intermittent cold exposure protocol that consists on basically exposing the mice to a very short cold exposure. The cold exposure, it's comprised by, during the first week, 5 minutes, during the second week, 7.5, during the third week, 10 minutes, and during the fourth week, 12.5 minutes of cold exposure. So the animal, it's stays on a normal facility on a 23, and then for these short minutes, it's daily exposed to a 6 Celsius cold exposure. And there we measured caloric intake and total weight weekly. And by the end of this intermittent cold exposure, we do the analysis, the functional analysis, the sample connection, and the analysis of the sample. Why did we venture on trying this intermittent cold exposure protocol? Because basically it allows us to have a physiologically and highly translatable protocol to activate brown adipose tissue and to try to study the impact of increasing thermogenesis in the humanized protocol. So let's start with the characterization of the model. So with this model, the mice, the intermittent cold exposure mice, they have an increase on the daily calorie intake, but they don't have a difference in the total body weight through the weeks. They do have a difference, a lower glucose levels. They don't have any difference in the THG levels and in the cholesterol levels. Because it's an exposure that the mice, they're not adjusted to the switch in the temperatures. The exposure to sudden temperature changes could, in fact, activate the HPA axis. That could lead to an increase in the stress of the mice. So one way to, we wanted to control for that. So we measured stress hormones, like we measure basically basal corticosterone levels. And as you can see here, there's not a significantly increase in the corticosterone levels of these mice. After the application of the cold exposure protocol, we measured tissue weights of white adipose tissue, inguinal and perigonadal, liver and muscle, and we didn't see any difference. But when we went and looked at the brown adipose tissue, we actually saw a difference, an increase in the amount of adipose tissue, of brown adipose tissue that we saw in ice mice. When we went and see the tissue, you can see, I think, pretty clearly in these pictures that the brown adipose tissue increased became basically more dark, more brown in this mice model. And also, if you compare these pictures and you can compare with the diagram on the left, you see that the brown adipose tissue is usually covered by some white adipose tissue. And you can very clearly see it in the control. That's pretty much lost in the intermittent cold exposure mice. When we see the histology of this brown adipose tissue, you can also see a very dramatic change in the morphology. You can see a reduction in the lipid droplet size. That could indicate a higher activation of the brown adipose tissue. Because we see such dramatic changes in the morphology, but also on the weight of the adipose tissue, we wanted to see if there was an increase in the differentiation of the brown adipose tissue. So we measured some differentiation markers, CBP alpha, and some brown adipose tissue differentiation markers, as I mentioned before, and there was an increase in all of them. So what we see is that actually the intermittent exposure protocol induces brown adipose tissue differentiation. We wanted to see also if there was an increase in the brown adipose tissue metabolic capacity. So we wanted to see if actually there was a difference in the amount of mitochondria that this brown adipose tissue was expressing. So we analyzed that by two ways. One by immunofluorescence, analyzing the amount of COX-4, some of the mitochondrial markers, and we saw an increase in COX-4 expression and also on UCP1. We also wanted to see if there was actually this tissue, besides being more brown and bigger in size, it was able to actually function better, oxidizing fatty acids. So we measured some of the fatty acid oxidation markers like LCAD, but also CD36. I think Simone mentioned it before, sort of a transporter that allows DAGs to get it into the tissue, and all of these markers were increased. So our next objective was actually to see if there was actually a thermogenic response to the cold, because you could have UCP1 expression, but that doesn't necessarily mean that it's functioning. So what we did is we did an experiment in which we took the mice that they were at 23. We exposed the mice for 12 and 12.5 minutes at 6, and we took thermal pictures. One thing that I didn't mention when I explained the protocol is that when we expose the mice to the cold exposure at 6, we need to individualize these mice. And so we control the individualization with a group that it's the control group that obviously we cannot use here, but here we have an additional group that is the naive group. This group, it's never been exposed to the intermittent cold, okay? So again, in this setup, we take naive mice that have never been exposed to cold and intermittent cold exposure mice that went through the whole protocol of intermittent cold exposure. And we take these two mice and we expose them at 6 degrees for 12.5 minutes. And hopefully you can see it as clearly as I do. The intermittent cold exposure mice is basically lights up. It's able to increase the thermogenesis much better than the naive mice. And we quantify actually the amount of heat from the brown fat and also from the inguinal and from the tail. The tail is only as a control. And we do see that there is an increase in the IBAT temperature and in the inguinal temperature as well. Then we wanted to see if there was actually basal thermogenesis improvement by this intermittent cold exposure protocol. So again, we have the naive mice that have never been handled and have never been exposed to anything. The control mice that, as I mentioned before, these mice, they went through the same process of the intermittent cold exposure, so they've been individualized for the same amount of time of the intermittent cold exposure, but they have never been exposed to cold. And these mice that they have been exposed to cold for the whole protocol. And as you can see here, the intermittent cold exposure mice, they have higher brown fat and white fat heat emission. So as you can see from the quantification and with the picture, the intermittent cold exposure is able not only to increase the capacity of the brown adipose tissue to do thermogenesis, but also impacts on inguinal. With that in mind, we wanted to see if this intermittent cold exposure could potentially induce browning of this depot. So what we did is first we analyzed the morphology of the inguinal white adipose tissue, and as you can see here, there is a reduction of the lipid droplet size and also an increase in the UCP1 expression that led us to believe that there's also some browning occurring there. And when we also measured mitochondrial markers like COX-4 or TOM20, we see an increase in both. So the intermittent cold exposure not only impacts on the brown adipose tissue, but also on the white adipose tissue, inducing some browning. So as a summary of the first part, we can say that the intermittent cold exposure induced some browning, increased the amount of brown adipose tissue, but also the capacity of the tissue to respond to a reduction in the temperature. And also the intermittent cold also induced the browning, in particular of the inguinal adipose tissue, overall increasing the capacity of the mice to sustain thermogenesis. So for my second part, we wanted to evaluate if the intermittent cold exposure protocol was actually, we could use it as a tool for metabolic disease. In particularly, we wanted to see if the intermittent cold exposure model could have a positive effect on the liver disease models. So the brown adipose tissue, it's basically considered like a metabolic sink. This actually means that it's able to incorporate lipids, carbohydrates, or also release lipids, basically depending on the need of the organism. So we wanted to harvest this capacity of the tissue to basically work as a metabolic sink and see if we could use it as a therapeutic tool. In the metabolic dysfunction associated steatotic liver disease, or MAFLD for short, it's one of the most prevalent liver disease in Latin America. It has like a 30 to 40% prevalence. It increases in visceral adipose tissue inflammation and in type 2 diabetes, and also has another set of factors such as poor nutrition and genetic factors. These two different causes could lead to the accumulation of lipids in the liver, leading to lipotoxicity and inflammation, oxidative stress, insulin resistance, and also the increase of collagen matrix. that over time could lead to inflammation, fibrosis, and cirrhosis. So we wanted to see basically if this intermittent cold exposure protocol, if it could be used as a therapy for this disease, basically targeting this part of the problem, which is the THG's accumulation in the liver. So in order to tackle this, we used two different models. The first model that we used was a high carbohydrate diet that consisted on adding 30% of sucrose in the drinking water of the mice and feed them normal chow. And we did that for 5 weeks. And then we measured if there was an alteration in the basal glucose levels. So as you can see here, the high carbohydrate diet had an impact on the weight of the mice, increasing the body weight. And by week 5 of this dietary intervention, there was an increase in the basal glucose levels. So with that, we started the intermittent cold exposure for another 4 weeks. Okay, so after these 4 weeks of intermittent cold exposure, we can see that it had a positive impact on the glucose levels, although it wasn't able to normalize completely the glucose levels to control. It was able to significantly reduce them compared to the high carbohydrate diet. Regarding the THG levels, the high carbohydrate diet induced the amount of THGs in the serum, but there was not a significant change with the intermittent cold exposure. And regarding the cholesterol levels, we pretty much saw the same thing as with the glucose levels. The high carbohydrate diet showed an increase in the cholesterol levels, and the intermittent cold exposure showed a partial reduction of it. Then we wanted to see if actually it worked as in the tissue that we were hoping for. So we went and see the liver, and we took some pictures of the liver. And as you can see, the high carbohydrate diet had an impact on the aspect of the liver. It became more pink, more filled with lipids, and the intermittent cold exposure kind of reversed that. You can see it much more clearly when you analyze the histology of the liver. The high carbohydrate diet induced an accumulation of lipids in the liver that was pretty much reversed by the intermittent cold exposure. And this was confirmed by the hepatic THG measurements that show that the intermittent cold exposure basically reduced very significantly the amount of THGs in the liver. Okay, then we switched to another model that was the high fat model that is 40% fat, and we pretty much did the same, but this time we treated the mice for 6 weeks with these dietary interventions. We saw that the mice gained weight, and that at week 6 there was an increase in the basal glucose levels. Thankfully, we saw the same, that the intermittent cold exposure was able to reduce the amount of glucose levels. Then we went and see if that had an impact on the brown adipose tissue, and it was able to actually increase the amount of adipose tissue or impact the morphology of the adipose tissue, we saw that the intermittent cold exposure had an impact on the morphology of the brown adipose tissue, reducing the lipid droplet size. And also we went ahead and we measured if there's actually an activation of this brown adipose tissue. So we did the same experiment at 23. We measured the basal activity of the white and brown adipose tissue. And this time we saw that there was actually an increase only on the brown adipose tissue depot. Then finally, we went and look at the liver, and here as well, you can see that the intermittent cold exposure also had an impact on the color of the liver. And when we went ahead and looked at the morphology of the liver, we saw that there was a reduction on the lipid droplet size and on the lipid droplet content without impacting the, this time, the hepatic cholesterol. Okay, so summary and conclusions. Hopefully I convinced you guys that brown fat is in fact a very important tissue, that it's highly metabolic, and it has a lot of potential and flexibility, that it's capable of increasing or decreasing the activity depending on the nutrient availability or the exposure of, in this case, the temperature, and that our protocol appears to be effective in on improving the metabolic fitness, and also could potentially be used as a therapeutic tool by increasing the thermogenic capacity and preventing or delaying the TAG accumulation in the muscle development. And finally, but not least, not the less important, this work was started and done mainly at the University of Buenos Aires, and then now sort of a migrant scientific that crosses the river pretty often. But I'm very grateful for my two labs, and also from the collaborators that are recently joined, in particularly Adali and Marina, and the star of this project, that is Joyce. So thank you all for your attention, and please ask ahead.
Thanks, Camila, for a nice presentation. Daliane already put hands up.
Yes, I have to talk and say hi, Camila. Hi, Carlos. Good to see you here. I didn't have the chance to say hello for you all, and I'm happy that you are here with us today. And nice presentation, Camila, quite interesting. And you have a very interesting model to study the reversion of metabolic flexibility and the adipose tissue dysfunction, looking at more specifically in adipose tissue, and it's very interesting. But it's a question from who don't work with a brown adipose tissue and cold. So
Sure.
Feel free if it's naive question, but I was wondering what would be the advantage of using intermittent exposure to cold instead of using continuous low temperature to induce brown adipose tissue activation. I'm saying that because here with our experience, if mice are in a low temperature environment, let's say 18 degrees, around 20 degrees, they do not develop obesity even when they are eating high fat diet, high carbohydrate diet. So I was wondering if you have some advantage of using intermittent cold exposure in order to prevent the chronic adaptation that may happen when you are all the time in a low temperature.
Basically, you just responded your question
Yeah,
yourself.
yes, I was wondering, so I'm not
Yes.
sure about that. And
Yes,
I was
by
only trying.
continuously exposure, first there are two things. One is exactly what you said. If you like cold exposure works, if you put the mice on a cold environment, they will do more thermogenesis. They will have higher energy expenditure, but also you will lose some sort of sensibility also to the ability of switching to between the switch on and switch off of thermogenesis. And the second that also I think it's very important is we think this model as a humanized model that it's able to be potentially adapted into a human setting. And I don't know about you, I really don't like cold, even though I work with cold. So I wouldn't dare to say a person, you know what, from now on, you need to put your AC at 18 for the rest of your life and basically live in a colder temperature. But you could ask a person that it's going through that has a metabolic disease or a metabolic disease associated with higher energy. You could say, you know what, if you take a cold shower for 5 minutes or 10 minutes, that will help you to increase your basal metabolic energy expenditure, and that could improve your the development or delay the development of your pathology. So
Yes,
basic. Yeah.
Yes, the last question is because I was wondering about the metabolic rate of those mice. Did you measure it?
I am doing those experiments. I am doing the energy expenditure of those experiments.
Yes, because to see whether it reflects in some change in whole body energy expenditure, it would be interesting to know. And another, because I was quite interested in what you showed,
Sure.
do you, about in a translational view, as you were, you said, taking a shower, a cold shower for 5 minutes. This is quite exciting. Do you know if someone have performed a clinical trial with this idea for humans?
I know there are a few, as far as I know, there are a few experiments that, for example, I think Camila Schill did on swimmers that go into winter swimming that showed improvements on some of the metabolic parameters. And I think there's one that they didn't show the results yet, but it's an ongoing protocol by the US Army that is actually testing this, but not for these short exposure times.
Yes,
Some
it
of
would
them,
be
I
quite
know.
interesting to perform it. And but okay, let's hear Carlos, because he was with a hand up as well. Thank you,
No,
Camila.
but I lowered the hand because I saw someone that was asking a question on the chat, and I didn't want to interfere with somebody else. I think that there was a question in the Zoom chat.
Yes, we can do the questions from Thierry. Nice presentation, thank you. How was the hepatic immune infiltration in intermittent cold exposure mice under diet-induced MAS LD condition?
Okay, thank you for that question. We're actually starting to analyze that. We have very preliminary data. That's why I didn't show anything. There seems to be a reduction on the, I think some of the interleukin-1 expression in the ICE mice compared to the non-treated one, but we just now started to study that. I don't have a really definitive answer on how this protocol it's impacting. We are more than happy to collaborate. We don't have the expertise. So as I show by my presentation, I'm more of a adipose tissue person. I use the liver as a readout. So we are more than happy to collaborate and study that, but we are just now starting to analyze that.
Understand.
And yeah, sorry about that. I don't have a good answer for it.
Thierry, I'd like to ask the other question that you made. So if you want. Or
yeah,
the
other question I had was about the progressive action of this ICE protocol. Do you think 1 week with a 12-minute exposure should be enough, or do you really have to go through this progression of 4 weeks, increasing the cold exposure to see the effect, the beneficial effect?
That's a great question. We haven't done those experiments, but what was the rationale behind the progressive cold exposure was to try to minimize the stress, because the mice, as they are continuously exposed to these 5 minutes, 7.5, 10, they obviously adjust to the handling and to the exposure. So going through the days, they are much more relaxed, so you can actually see the impact on only the temperature and not the stress of the mice. That's why we wanted to also do it progressively.
Yeah, I was wondering, considering the corticosterone levels you were so maybe there's at some point after 4 weeks a decrease in corticosterone levels because some kind of habituation or...
Yeah, that could be. That could absolutely be. Also, to be 100% fair, we only measured basal corticosterone. We didn't do like a ACTH.
Like would be nice to see, yes.
Yes, of course. But we only did the basal corticosterone, and we did it in the normal diet, but also in the
We lost Camilla.
Yeah.
I think we lost Camilla, yes.
You're back.
Oh, sorry.
You're back.
Okay.
I
warned. Sorry. I don't know if I answered your question. Okay, thank you.
So because of the time, we will move forward. And thank you, Camila, again for your presentation.
Thank you.
To close our session today, we have a distinguished guest, Dr. Carlos Escander. Carlos is the principal investigator and head of the Laboratory of Metabolic Diseases and Aging at the Institute Pasteur de Montevideo. His journey took him from a PhD in Uruguay to a postdoctoral fellowship at the Mayo Clinic Alex School of Medicine in the US. Beyond academia, also Carlos is an innovator in the biotech space as a co-founder of Eolo Pharma, developing therapies for metabolic disorders. It's a privilege to have you here with us, Carlos, and please go ahead.
Thank you. Do you see my screen? Is it in full presentation?
No, we see that one that is black.
Oh, sorry.
Black
I need to stop sharing. Just give me 1 second. There we go. I think I should be. Do you see the right
Now
one
it's
now?
okay. Yeah.
Okay, thank you. Sorry for that. So let me look for the pointer. I may need it. So thanks, Marina and Adaliene for inviting for this presentation. I'm gonna talk about this drug development that we have been doing here in Uruguay in collaboration with many countries and labs around the world and with a lot of people in Brazil also. And I have decided, knowing that there was gonna be a lot of people talking about adipose tissue function, and also about obesity and brown adipose tissue, as Camila was talking right now. I decided to focus more about the journey that we have been walking in the past years and how we got here. So I'm gonna tell how the story developed basically, and I hope you will enjoy. And this is my message, especially for young people, about this talk will be that you need to follow the science, and many times the most amazing results come very unexpected, and it's not what you are looking for. So this would be my main message that I will try to show it. So there, very quickly, I do have to declare a conflict of interest because besides, as Marina was saying, besides being the head of the lab here at Pasteur Institute, I also founder and I have shares in this small company that actually is a company that is based at Pasteur also. So most of the people that work in the company, they are scientists that are involved in basic research at Institute Pasteur. And I was saying, so the story is gonna come from a very simple idea that we had almost 10 years ago. This I'm showing you here. This is a picture of the Amazon River, and we were planning to develop some drugs, and the idea was very straightforward. But the truth is that the reality was very different. So we were able to go from A to B, but the journey was a little bit more complicated than what we expected, but actually was much more exciting. So we are very happy that we finally got from A to B. It took longer than we expected, but we finally got there. So I'm gonna go very quickly in the introduction because we had very nice talks about obesity and the consequences of that and all that. So basically the trigger for this was the constant growth of obesity and obesity-related disorders about all over the world. We can say that malnutrition is one of the biggest issues in the whole world. Most countries that do not have overnutrition, they do have undernutrition. So nutrition is a big issue in most countries, and it has been growing non-stop since the 60s and 70s, and in Uruguay and in Brazil and Argentina, the numbers are also very preoccupying. I mean, we have in Uruguay 65% of the population, and 40% of our kids are either overweight or obese. So it's a big health issue in our countries also. And we all know that, I mean, the main one of the main problems with obesity, besides obesity itself, is the prevalence of the incidence of many comorbidities, especially type 2 diabetes, that they are calculated that more than 400 million people they suffer from type 2 diabetes, and the main trigger for that is obesity. And of course we all know that, I mean, when we started doing this, the GLP-1 receptor agonists were not in the market, at least not for obesity. There was liraglutide was being tested for type 2 diabetes, but there is a huge revolution involving the GLP-1 and Ozempic and all that, And there is a lot of consumption. I mean, and the results they have been amazing in terms of body weight reduction. But we all know that they have a lot of side effects. There is a lot of concern about some of the side effects, especially in long-term consumption. And I would say that sarcopenia, muscle wasting is one of the main concerns, especially in the elderly. The loss of muscle mass is a big concern. And there are also some issues regarding that how expensive these drugs are. And so I would say that it is very clear now that obesity can be pharmacologically treated, something that until a few years ago was more of a utopic thing, basically because there was a huge story of failures in treating obesity from the pharmacological side. But everybody knows that the treatments either for those people that has been already identified that are resistant to the GLP-1 receptor agonist, or for the side effects. The pharmacological approaches are still needed, and actually the big pharma companies are looking for alternatives for either combinations or alternatives for the GLP-1 treatments. So we started this story under this paradigm that if we could tackle chronic inflammation during obesity, and we all know that inflammation is a strong force in the developing in the onset of and developing of metabolic comorbidities during obesity. So the idea that started our work, and this was more than 10 years ago, was if we can pharmacological targets for inflammation, chronic inflammation during obesity, we should be able to target most of the comorbidities. And the story started from these small molecules, the nitroconjugated fatty acids that were developed mainly by the group of Bruce Spiegelman at the University of Pittsburgh, with some of the people in our team trained a few years back. And basically what they showed in Bruce Freeman's lab was that if this small chemical part of the conjugated fatty acid, this nitroalkene, was able to react chemically especially with cysteine and histidines in proteins, and also with glutathione, and generate different cellular responses, especially having anti-inflammatory effects. And it was, I mean, there's a lot of research done in this. Actually, some of these molecules went into clinical trials. Some are still under investigation in patients, and for some of these chronic diseases that I have a list here, there were some very promising results. But there were some issues with the pharmacology of these molecules, especially metabolization was a problem. They are very unstable. The long chain fatty acids, they are rapidly metabolized by mitochondria. They are saturated. So there are many downsides by the approach, but we were very excited about the chemistry and the biology of the nitrogen group. So the idea that we developed in our research group, and one of them, this one chemist and one medical doctor, that the three of us were the team that started with this idea, is that okay, let's take this chemical this nitrogen group and put it into different molecular scaffolds, because the idea was that the main problem with the nitric fatty acids was not the nitrated part itself, but also the scaffold. So we say, okay, let's try to move this nitrogen group to different scaffolds and see what happens. And the idea here was that we were going to be able to decrease inflammation, and by decreasing inflammation, we were going to be able to tackle different aspects of metabolic syndrome during obesity. This is the first drug that we developed. It was a nitrile king analog of vitamin E. This is the alpha tocopherol, the original molecule of alpha tocopherol. The native one is the one in black, and then what the thing that we did was to add this nitrogen group to the alpha tocopherol, and we tested this in a model of atherosclerosis, and we showed this was 7 years ago. This is the first papers we published with this idea, was that if we fed ApoE knockout mice with high fat diet, we induced plaque formation in the aortas. Here, what you see, the plaque formation stained with a lipid dye is the whole aorta dissected, and each one of these is a plaque. We showed that there was a decrease in plaque formation in the mice, and actually that there was less inflammation in the plaques. There was also decreased inflammation, and this was I always like to tell this part of the story, because this was the founding idea of what happens later, and it was that we could easily relocate this group and it was still functional, and that we could use the molecular scaffold either to provide to direct the nitrogen group to a specific site. For example, in the case of alpha tocopherol, send it in with the lipoproteins to the site of the plaques, or to use some other characteristics of the scaffold that could help us to develop better drugs. The second proof of concept was to develop a more soluble form of the nitro of the vitamin E that we call NATXO, and we published that in a context of diet-induced obesity, we were able to reduce inflammation, and actually we showed that this drug would inhibit the inflammasome. And although the mice under diet-induced obesity, they become obese, they were protected against metabolic dysregulation. So and this was also important for two things. One was because we showed a second molecule that had a different scaffold, that had very interesting properties as an anti-inflammatory molecule that could tackle chronic inflammation during obesity, and second, because it was a molecule that we could patent and protect, and this was the trigger for a parallel journey that we started, that was to start a small company that allowed us to bring money in order to keep our developments going and to move to our studies to the idea was to be able to go into clinical studies, that, as I will show you later, was what we finally did. But this molecule allowed us to show that we could generate novel molecules that could have an effect, at least in preclinical models, tackling inflammation. But we were still not very happy with some of the results in terms of the potency of the molecule in these two molecules, and there were some things about the pharmacokinetics and biodistribution that didn't were not very convincing to us to move forward. So we went for a third generation of drugs, and we decided to choose this small molecule. Is probably the oldest known drug in humanity, that is salicylate. As you probably know, salicylate is the precursor of aspirin, has been used for centuries, and actually there is the first record from the use of salicylate derivatives is coming from the old Egyptians, the Ebers papyrus. And the salicylate in the form of aspirin has been used for more than a century for chronic consumption. The safety profile is very well known, and we wanted to build our new drugs on molecular scaffolds that we knew that they were not going to be toxic in a human setup, because The idea that we were pursuing is try to develop the best possible drug for clinical development. And we all know that being in South America, we wouldn't have a second chance to succeed. So we wanted to make sure that if we were gonna try to take this to the clinic, at least the molecule was the safest possible that we could manage. And there were, as I was saying, salicylate has been taken daily, is still taken daily by half of the adult population in the United States. Although there are many other like ibuprofen has displaced the use of aspirin for headaches and all that, is still heavily consumed, at least in the United States. So and there was early in the 2000s there was like a rediscovery of salicylate. It was shown in a paper in Science in '94 that both salicylate and aspirin were inhibitors of NF-κB. And there were some clinical trials developed in the later between 2010 2018 showing that actually salicylate had nice effects in patients with obesity and type 2 diabetes. The problem with this was first that the effects were not better than metformin, and metformin was in first line drug for the treatment of type 2 diabetes. And second, that the doses that were necessary to have an effect were extremely high, were grams of salicylate taken daily by the patients that were needed to see an amelioration in metabolic syndrome. So although the trials had positive results, this development was discontinued. But based on all that, we decided to synthesize what it became our leading molecule that we call SANA, that actually, as you can see here, is salicylate, where we added the nitroalkene in this carbon here. And this is so as I'm telling you guys, so what we were looking for was to test the molecule in an obesity setup, and the focus was trying to go after chronic inflammation. So the idea was, okay, we are gonna get our mice on a high-fat diet. The mice are gonna become obese. We're gonna treat the mice, and then we're gonna see obese mice, but they are gonna be healthier. This was our hypothesis, and this was since the beginning was developed by Karine. That she did an amazing job in developing this project. And the first experiment that she did, again, we had a plan, we had a hypothesis, and then we came into Narnia. Okay, so there was completely turned around, because when she started feeding the mice with high-fat diet and SANA, what she saw was that the mice were not gaining weight. And it came as a huge surprise to us, because, I mean, we were not expecting this. Actually, we were, for quite some time, very worried about the results, because I was a little bit skeptical that maybe there was some unwanted effect, let's say toxicity, or that the mice were eating less, that there was, I mean, I was very, very worried about the fact that the mice were not gaining weight. But the protection, as you can see here, this is a dose response for SANA treatments. These are the control mice. These are the mice on a normal diet, mice on high-fat diet, a dose response with SANA. And actually the mice on 400 milligrams per K of SANA, they did not weight gain at all. They were completely lean. And we had these mice for months on a high-fat diet, and they didn't gain weight, and we never saw any sign of toxicity with the mice. We, of course, every single time we use salicylate as a control, the precursor of the drug, and at very high doses, you see some effect of salicylate that has been described, but it was nothing compared with the effect of SANA. And from the beginning, we knew that the effect of SANA in preventing obesity, of course, was the decrease in fat tissue accumulation, and it was very, very clear. Opening the mice, the mice had less fat, and the rest of the tissues looked very, very, very normal. Actually, if you put one of these mouse double blind with a mouse on normal diet, it's almost impossible to differentiate the control mouse with the mouse on a high-fat diet and treated with SANA. We extensively measured food intake, and we saw that there were no changes in food intake. And basically what we were seeing was, okay, we prevent obesity, and every single thing that we could measure in terms of metabolic dysregulation caused by obesity was prevented when we treated the mice with SANA. And I'm showing just a summary of the results: fasting glucose, glucose tolerance, liver steatosis. We collected a lot of data on liver damage. I mean, every single thing that we measured was prevented by SANA. But then the next challenge was to see, and there have been plenty of drugs published in mice treated under high-fat diet that they work really well in prevention of obesity, but then you treat the mice and you don't see any effect. So we went for a protocol of treatment of obesity. So we let the mice to become obese, and after they were obese, after 5 weeks, and after the glucose levels were altered compared to mice on normal chow, we started treating with SANA, and we saw very nice effect in weight loss. And this was not due to changes in food consumption. I mean, we measured every single time food consumption, and the food consumption was the same. The mice were eating the same amount of food corrected by body weight, and they were losing weight. And again, not only they were losing weight, but their blood glucose was corrected, glucose tolerance, free fatty acids, liver steatosis, markers of liver damage, they were also improved under this protocol of treatment of obesity and obesity-related metabolic dysregulation. We compared with metformin, and we saw that again, so that we see a loss in body weight when we treat the mice with SANA, not with metformin. That is the one in blue. When we measured glucose levels, basal glucose levels in blood before and after the treatments, we saw that the mice on a high-fat diet, the glycemia increased. Salicylate and metformin, what they did was to stop the increase in glycemia during treatment, but SANA alone was able to decrease basal glucose levels. The combination had no effect. So the dominant effect, the combination with metformin, so the effect was driven by the SANA treatment. Next, and this is unpublished, we moved to compare the treatment between SANA and GLP-1 receptor agonist, and we did a comparison for liraglutide and semaglutide, and we see exactly the same result, that if we treat with low doses of GLP-1 receptor agonist or with SANA, we can see a decrease in body weight when the mice are obese, but when we treat with the combination, there is a massive decrease in body weight. And we have been scaling down the dose. I'm not showing the results, but now we know that we can, if we treat the mice, the obese mice, either with SANA or with Ozempic at doses that are not effective by a lone treatment, when the drug is given alone. When we give the combination, we see a reduction in body weight. So we are very excited about the prospect that this could be part of a combo treatment to either decrease the effective dose of the GLP-1 receptor agonist, or either to do a replacement treatment or something like that. But we are working on that. So the main challenge that we faced and was when we saw the results, and we were very clear about the phenotype. We knew that there was prevention against obesity. We knew that the obesity-related metabolic dysregulation was being corrected by SANA, but we had no idea about how SANA was working, and we spent a lot of time trying to determine this. And it's something that we learned is that it's very difficult to go from a phenotype into a molecular mechanism of action. And actually, we are still trying to understand exactly how this is working. But basically, when we decided to go after the trying to understand how SANA was working, the reasoning was very simple. So obesity comes from a disbalance between calorie intake and energy expenditure. We knew that calorie intake was the same, and we knew that we measured the amount of feces. We knew that there was no diarrhea. We knew that absorption was okay. And we measured every single aspect regarding to digestion of the diet or the drug, and we didn't see any difference. We measured the calories in the stools, everything that we could measure. We did, and we knew that there was no, I mean, the amount of calories that were going in were the same. The amount of calories that were being excreted were the same. So the only thing that was left was energy expenditure. So this could be because the mice are doing more exercise. Energy expenditure can be achieved by running more, by moving more, or it could be resting energy expenditure. So we measured resting energy expenditure, and we saw that, and this was done by indirect calorimetry, and we saw that just a few days of treatment with SANA was able to I wouldn't say dramatically, but to significantly increase energy expenditure. So, and we knew that it was resting energy expenditure because we measured movement, we measured muscle function, and we rule out muscle as a player in these changes in energy expenditure. So we went to measure, as Camila was explaining, thermogenesis. We took these pictures with an infrared camera, and you can see here that the mice treated with SANA, they are producing more heat. So the next thing was to measure if the stimulation with the beta-3 specific agonist CL316 that was introduced before today could show a differential response between naive mice and mice treated with SANA. And again, when we treated the mice with CL and measure energy expenditure by indirect calorimetry, we saw that CL was increasing more energy expenditure than mice when the mice were treated with SANA than in the control mice. So we came from these results and analysis of the results that I'm not showing to the conclusion that what SANA was doing was increasing energy expenditure in adipose tissue and promoting thermogenesis. I don't need to explain this because Camila just gave a great talk about how adipose tissue works and how brown adipose tissue works. But what was really important for us was, okay, that brown adipose tissue is present in humans, in adult humans, is functional in humans even under physiological conditions. And I'm showing here one of the papers. This was shown in 2009. The same individual. This is FDG uptake and in the winter, in the summer, showing that the amount of FDG uptake in these depots of brown adipose tissue is increased between summer and winter. So we know that this old paradigm that brown adipose tissue is lost in adults is no longer true, and we know from Camila's talk that white adipose tissue can be converted to beige fat if it's stimulated. So as I was saying, we knew that this, we were convinced that this was energy expenditure, that it was activation of adipose tissue. The next thing that we tried to sort out was to determine the mechanism of action, which mechanism was being involved in increasing energy expenditure and heat production. And of course, the first thing we did was to go after UCP1 activation. And I can tell you, we spent months trying to show that what we were seeing was increase in UCP1 expression or UCP1 activity. And there was no way that we could see an increase in UCP1 expression or activity. And actually, what we repeatedly saw when we analyzed UCP1 expression was a decrease in UCP1 expression when we treated the mice with SANA. At the same time, and here I'm sorry, I forgot about this. And not only that, when we treated the UCP1 knockout mice with SANA, here I'm showing obese mice, wild type and knockout, UCP1 knockout treated with SANA. These are in black the control mice, wild type and knockout. And when we treat wild type with SANA, we see a decrease in body weight, but we see the same in the UCP1 knockout mice. So we have pretty good evidence that this was independent of UCP1. There are other mechanisms of thermogenesis, and this is a review from Lawrence Kazak that was mentioned before by Simon, that he has been working really hard in identifying new molecular mechanisms of heat production and thermogenesis. So, but we at that point, and this when we were doing these experiments, Lawrence's paper had just came out in Cell, the first paper. So we had no idea what was going on. We didn't understand how our adipose tissue was being activated. So we took advantage that we have a very cool mass spec facility here at Institut Pasteur. So we decided to take the inguinal white adipose tissue and do proteomics on the mice treated with either control diet or a high-fat diet or high-fat diet like SANA. And the results that we got from this proteomic analysis was amazing, and showed us the first fingerprints pointing to creatine-dependent thermogenesis as the mechanism of action of our drug. So we detected we started seeing some creatine kinases that were being overexpressed in adipose tissue, and other markers of creatine metabolism and creatine-dependent thermogenesis. And to make a long story short, we were able to show that there is some molecular fingerprint of creatine-dependent thermogenesis, both in white adipose tissue and also in brown adipose tissue. There is an increasing creatine synthesis, there is an increasing creatine kinase activity. We do not see the same creatine kinases upregulated with SANA that one sees with cold exposure. So what we think that SANA is mimicking creatine-dependent thermogenesis, but it's not activating CKB expression, it's activating creatine kinase, the mitochondrial creatine kinase expression, and also another cytosolic creatine kinase expression. We still do not understand why is that, but it seems that SANA is phenocopying the creatine-dependent thermogenesis, but it's not exactly it's not mirroring cold elicited creatine-dependent thermogenesis. We still don't understand why this is happening and how this is happening, but we are pretty sure about our molecular mechanism of action of SANA. And it was very interesting, and we got so this is SANA, the molecule of SANA. So you can see here that the nitrogen group was added in the carbon 5. The reviewers in when we sent the paper, they wanted to show how specific SANA was, because it was hard to believe that we got lucky and SANA was so specific, and actually we had to develop different isomers of SANA. It's exactly the same chemical formula, but we had to move the nitrogen group to different carbons, and in every other position that we put the nitrogen group, the effect was completely lost in activation of thermogenesis and creatine-dependent thermogenesis. So we are still trying to understand why this molecule is so specific for this and how this is happening, and we have been working night and day to try to understand the molecular target of the drug. It's still elusive, but we are putting a lot of effort on that. So and I'm gonna take a few more minutes. Do I have time, Adeliene? Yes? Okay. So just a few more minutes to show how we went from this very nice, I'm being, maybe somebody else needs to be that is very nice, but we are very proud of the work. So what we believe was a very nice work in a mouse setup in the context of diet-induced obesity, to this crazy idea that we had when we started, that was trying to get our small molecules to the clinic. Yes, and so this is just shown by an arrow to go from mice to human. It was a huge journey. So we had to run a lot of tox studies. I mean, it was an amazing amount of work that we had to do. We had to raise a lot of money because this is not something that can be done in a lab. It has to be done by certified companies, third parties. So it was very, very complicated. It was a very complex journey that led us to get the approval to develop a clinical trial and see if the drug could be, the plan was to test if the drug was safe and what could be tolerated by humans. So we developed this clinical trial that was developed in Australia. So basically we divided in two phases, phase 1A and phase 1B. The phase 1A was single ascending dose in healthy volunteers. And for the phase 1B, we decided to go a little further. And although the main goal of the trial was to test safety and tolerability, we decided to go for a multiple ascending dose in obese or overweight volunteers and see if not only if the drug was well tolerated and was safe, but also to see if we could detect any kind of signs of that the drug was being effective. So this is the scheme of the single ascending dose and multiple ascending dose. We went from 200 milligrams to 800 milligrams in the single ascending dose. In the 800 milligram dose, we had some a renal effect in one patient that was when the drug was discontinued, it was self-corrected, so nothing major, but this led us to eliminate this dose, and then for the multiple ascending dose, we did 200 milligrams and 100 milligrams, and then 150 and 200 milligrams twice a day for 2 weeks. And just showing the effective effect of the drug. The drug was well tolerated, was safe. There were no the only side effects that there were very, very mild, and actually were also present in most of the placebos. So there was only one side effect that could be attributed to the drug, and it was mild, so that it was some diarrhea. So in terms of tolerability and safety, the drug did very, very well. But not only that, but we also saw some decrease in body weight. Of course, you can see that we have a very small amount of patients, so we have to be very careful about the interpretation of this, and we are very happy, but it was in a very small group of volunteers. But not only that, we saw, except from one volunteer, we saw an improvement in glucose levels. In all patients, we saw a decrease in insulin levels in serum. In insulin resistance measured by IR test was also significantly improved, and also fructosamine that we measured because of the half-life of albumin is less than hemoglobin. We measured fructosamine, and we saw also a decrease in glycated albumin. So and this was after a long work and many different things that we had to respond was finally accepted in Nature Metabolism a few months ago, and we are very happy about this. So we can say that we were able to go and from the bench, because actually the drug was designed in the lab and was first synthesized in the lab into a clinical trial, and this is, as far as we know, it's the first time that this path is covered in South America, going from the design of a drug, a synthesis of a drug, go through all the preclinical studies, and finally get to phase 1 clinical trial. We're trying to start a phase 2 clinical trial that now we are gonna stay in South America. So we already have signed to run a phase 2 clinical trial that is gonna be more than 100 volunteers. That is gonna be done in Chile. It's starting early 2026, and we are hoping that we are gonna be able to show that not only that SANA is safe and well tolerated in long-term treatment. The idea is to go to 3 months and maybe 6 months if everything goes well, but also to show that it's effective in a bigger cohort. And I think I'm gonna skip this because probably I'm already on time. So I'm gonna skip this and go to the acknowledgements. So this is a huge effort that was I mean, a lot of people participated. As I mentioned before, Karine was the leading author of this work, together with another guy, Alejandro, that was the one that did all the proteomics. There are many groups here at Pasteur and Universidad de la República here. We had a lot of collaboration from Brazilian groups, people in Unicamp, in USP. I forgot in this presentation to put the people from Herédia that were also involved in this work. I didn't show the data, but they did a lot of work in mitochondria in the Department of Biochemistry in Herédia. And actually, we did a lot of work with my former advisor, Eduardo Chini, that he's also Brazilian. He's from Niterói, but he has been for many years in the US. But his heart, I think, is still in Rio. So I think it's a very nice example of how we can do great stuff if we work together as teams, and if we follow the science and not our hypothesis, because our hypothesis was completely wrong regarding to SAN. And here's the team. So Karina, the first author of the paper, together with Alejandro, and Camila is now doing an amazing job in the liver, not only with SANA, but also with how cold exposure reshapes the liver function to sustain thermogenesis during cold exposure. So thank you very much, and if you have any questions, I'm very happy to answer them.
Thank you so much for this amazing, wonderful history. It was really beautiful to see from where to now where you are, and the future. It has a lot of possibilities, a lot of things that still can be done. So it's amazing. I will start with a question. You said that you worked with SANA in different tissues. You demonstrated liver, adipose tissue, but in muscle.
In muscle, it's muscle. We have. I mean, we in muscle we measured so far the things that were necessary to convince the reviewers that the driving force in our setup was adipose tissue. But we measured. I mean, we measured the direct action of SANA on muscle, mitochondria, and we did respirometry in muscle fibers, both in mice treated with SANA and also in isolated fibers, and we never saw an effect in terms of muscle function. We measured muscle contraction during cold exposure, and we saw that the Shivan thermogenesis, if we treat mice with SANA, when we treat mice with SANA, they do better in a cold exposure. And when we measure shivering thermogenesis, we saw that the muscle was contracting less. And our interpretation was that they needed less muscle heat production because the adipose tissue was already helping thermoregulate. And I don't know if Camila is still around, but Camila has some preliminary data that, and in her protocol also, when the mice seem to be doing less shivering during cold exposure. With that being said, we also measured creatine levels in muscle, and we saw increased creatine levels. We don't know yet if this could be a reflect of changes in muscle function that we didn't measure in the right way, or if it's because creatine metabolism is upregulated in the whole body, and we measure creatine levels in adipose tissue, and we saw increased levels of creatine in adipose tissue, that maybe this turns into a systemic upregulation of creatine metabolism. We don't know. In the liver, we also saw it was not significant, but there was a clear trend to increase the creatine levels also. And we are working in trying to understand how the liver is playing a role, because we are convinced that the liver is playing a key role in some of the SANA effects, even in adipose tissue. But other than that, we don't know, and I think that this would be very, very cool to see what is, to focus on muscle and see what is going on. We have a collaborator now in England that is starting to measure some metabolites in the heart to see what is going on in the heart also. But we don't know more than what I said, but I think it's very interesting. We know that, we measure muscle function in many different ways. We put the mice on treadmills. We measured a lot of things in terms of function, and we didn't see improvement, but we didn't see any deleterious effect. Other than that, I don't know.
But have you measured, for example, the body composition of these mice, in the sense to see how it happens with SANA?
It's a good question. We measured by MRI lean mass, and the total amount of lean mass was not significantly changed in the conditions that we measured. Yes. Now we are and I'm gonna be very cautious on this, because the evidence that we have is still very preliminary. We have some evidence that if we treat obese mice with SANA, there seems to be an improvement in muscle function, but we don't know muscle mass. We only have muscle function, and we have in a certain experimental setup, and we only did a certain type of measurements, and we need to be cautious about we are very excited about this, because actually we saw that they did much better than the mice treated with Ozempic, and we think that it could be that SANA may help prevent muscle wasting during GLP-1 treatment, but it's very early to tell. So I think there is a whole thing to explore there. Yeah.
Okay, thanks. I have here another question from Amanda that presented us before. What do you think that SANA does in the fibrosis in adipose tissue?
Oh, this is a very interesting question. I'm hoping, and we haven't. If you do a treatment of prevention, the adipose tissue looks like a tissue on normal diet. You don't see anything. You don't see any fibrosis. But we don't know what is going on under the protocol of treatment. We have data suggesting that inflammation is decreased. We are hoping that Joyce will be able to show that there is decreased senescence. We have some evidence in vitro, and we haven't done anything in vivo yet. We have some evidence in vitro that SANA could be acting not as a partial suppressor of the SASP. Would be not I'm losing the word now. It's not a cell that kills senescent cells, but is but that decreases the inflammatory phenotype. This is in vitro, and we think could be related to the anti-inflammatory actions of SANA. But my hypothesis, if I had to say, would be that there should be some decrease in or amelioration of fibrosis. But this has to be shown, and I have been wrong many times with all this, so we have to go and check.
Hi, Carlos. Just to finish, thanks. It was really nice to hear you. So, do you think that SANA indeed maybe because you say that you don't have data, that maybe SANA targets the fibrosis synthesis in obesity?
This is you are taking me to a place where I don't feel very comfortable, but I would say no, because I don't have a clear answer to give. What I would say with some confidence is that it should decrease some of the triggers of inflammation and fibrosis.
Got it.
But no more than that.
Okay.
Sorry?
The idea the idea that of what Joyce is doing, that maybe SANA works in the targeting the SASP, and since fibrosis is
Yes,
like
we are finishing a manuscript right now that we are not 100% sure that this is gonna be the main driver, but of the effect that we see in adipose tissue. But we know that something that happens in terms of inflammation is that when you treat mice with SANA in the very short term high fat diet, and we took all this idea of going into short term studies from Adaliene to see what was going on at the very beginning of the treatment. What we see is that, and we only have clear data for macrophages. We see that the macrophages are infiltrating, but they are not being able to fully polarize to M1. And it seems that the monocytes go in, they get to the place when you put them on a high fat diet, but they are not being able to fully differentiate. And actually, we did the opposite of treating the mice with SANA, looking for increasing energy expenditure and injecting M1 macrophages into adipose tissue. And it seems that when we inject in vitro differentiated macrophages with interferon into the adipose tissue, when the mice are doing more thermogenesis, it seems that thermogenesis is shut down. So if this implies that the crosstalk between macrophages and adipose cells is the target or is the main driver of the SANA action, I don't know, and I'm very careful about that. But we are following this idea that maybe because something that is very clear in our experiments is that the effect of SANA is very, very early. It's extremely early, and actually we see the increase in energy expenditure way before we see the increase in weight gain. So we think that might be something very, very some very early event, and we are going after these very fast immune infiltrations that are not the consequence of the obese mouse or the already hypertrophic adipocytes and all that, but instead of that, these very early responses that you guys know way better than me.
Okay, got it. Thank you so much, Carlos.
No, thank you.
Now, Adaline would like to say something.
I have to.
Are you going to kill me from?
No, no.
You can.
I promise, Marina, that I wouldn't talk, but I can't. I would like to discuss, but it's just a comment. First, it's amazing your history in science, Carlos. It's inspire us, not only the oldest research, but but the youngest one. So thank you so much. Amazing. But have you looked for the pro-resolution molecules because as you said, you don't have a shift between M2 AM1. Maybe because SANA has a lipid feature, no?
Mm-hmm.
And we have a lot of molecules that are pro-resolutive molecules.
Yes.
That has a lipid chain. I don't know, maybe resolution of inflammation should be an answer, not exactly for the inflammation by itself, looking for pro-inflammatory cytokines or anti-inflammatory cytokines, but go further to check the pro-inflammatory, pro-resolutive cytokines, including annexin A1 and so on. Just an idea that maybe we could.
No, yes, I mean, and we are 100% open to discuss and to collaborate and to receive help, because actually we I mean, my knowledge in immunology is the textbook knowledge, and we always struggle when we get to this area of the crosstalk between adipose cells and immune cells, and it's so complicated.
Yes.
So complicated that
But we
and
have
it's
to
hard
take a note.
Yeah, at least in our experience has been very, very hard to try to pinpoint a single pathway or a single cell. Now we are putting a lot of effort in see if we can do single cell RNA-seq to see if we can get some clear signal from the whole landscape from different cells to see if there is something that we are missing in the synaption. And because we went after macrophages because we knew that there was some effect on macrophages, and we knew I mean, and the tools to study macrophages, but we are being almost blind to any other kind of immune cell that could be either playing a role, cross-talking. I mean, it's extremely complex. So there's a lot of things to do there. And actually, we didn't lead this work, but another group here in Pasteur that were collaborators of us, they showed that SANA plays a key role in immune response in the context of graft survival, and they regulate T cells. I mean, there is, and this is how we decided to jump into the immunometabolic crosstalk, because we have the impression that and the impression, and there is data from this other group that SANA in different contexts may be playing an immunomodulatory role
Modulation.
yeah.
Amazing. Okay, thank you, Carlos. Thanks so much.
No, thank you guys.
Oh, now you finished? After all of these amazing talks, and thanks, Carlos, for the last one, but was really amazing. Before we say goodbye, for the final closing remarks, please come to screen our group leader and organizer, Adaliene.
Yes, I'm just would like to, as we are bringing to the end of the successful second scientific meeting of immunometabolism, I believe we can all agree that the dialogue shared over the past sections have been highly stimulating and profoundly productive. I wish to extend my deepest gratitude to all our distinguished speakers. Your cutting-edge presentation from the complexities of immunometabolism to the frontiers of clinical translation have been truly inspiring. We offer special thanks to the international partners. This meeting has been a vibrant expression of our collaborative objectives, particularly those supported by CAPES-COFECUB and the CNPq International Program. Your commitment is the foundation of our progress. Our thanks go also to the long-standing partners, Geneviève Marcelon and Karine Clément, to their sustained support and for fostering our path for a collaborative thought. We also sincerely thank Dr. Carlos, Escant, Camila, Carla, and our dedicated Brazilian collaborators from UFMG and UERJ for ensuring the continuity of this intriguing research journey. A great thanks goes to every attendee for your active engagement and to the organizing committee for the brilliant execution of this event. I cannot forget to say thank you so much for the immunometabolism group that have been working together to make it happen. Thank you so much. The field of immunometabolism demands the scientific rigor and collaborative spirit we have here, seeing that how important it is. We are confident that the connections made will fuel the next generation of students and researchers. So we officially conclude the second scientific meeting, and thank you all again. And we look forward to welcome you all for the third version of our scientific meeting, I hope in a near future. So thank you so much for all of you, and hope to